First of all, I must welcome all the friends who are joining us from all over the world. I am so happy to know that people all over the world are excited to learn from each other, especially during these bad times of Corona, where we are all locked into our houses and our minds and our thoughts playing tricks with us. But I guess you see, the human mind is the best example of yearning to learn more, to bypass the situation, adapt to bad situations, and survive and thrive. So I guess this Corona times is seeing us through a lot of learning experience. We are having a lot of these fantastic webinars. I am so excited to come across people from all over the world. We have guests joining in from Germany, Philippines, Thailand, Malaysia, Switzerland, Middle East, Spain, and even as far as Turkey. So I was so overwhelmed to see the response of the questions coming in. They were so intriguing questions, thought-provoking questions. And I think it is going to be a great session. And I must welcome my dear friends, Dr. Himanshu Matalia, who is there. He is a very, very senior and a renowned surgeon all over across the country and an international renowned speaker. He's a very senior cornea surgeon and a cornea refractive surgeon. He's a medical superintendent of one of the top eye institutions of the country, Narayana Nethralaya. He's a medical superintendent of Narayana Nethralaya too. And he's a fantastic speaker, as you all will experience him. And on my right of the screen is Dr. Sonu Goyal, again, a very dear friend, an exceptional surgeon, a prolific orator, a beautiful teacher, and we all have shared stages all over the world and national and international conferences. Dr. Sonu Goyal is the managing director of Anand Eye Hospital in Jaipur, Rajasthan, a very, very, very renowned high volume surgeon. Sonu again comes with a baggage of a lot of high volume surgeries. So all three of us together will try and pool in our knowledge of phakic IOLs. And I guess between all of us, I wouldn't be wrong, Sonu and Himanshu, approximately 10 years of phakic IOL experience, so 30 years of phakic IOL experience poured into a funnel, and we will try and make a concentrate of that. Huge learning is going to be there in this session. There are going to be a lot of questions answered, a lot of myths busted, a lot of surgical videos, a lot of complication management to be seen. But in the end, once the session is over, we will see that we all go back enriched. Even it's an opportunity for me to learn from Himanshu and Sonu and vice versa. So similarly, I guess we should all utilize this opportunity to learn from each other. A few words from Dr. Sonu first. Dr. Sonu.
Thank you, Kamal, for making me part of your wonderful course. And I'm so very overwhelmed looking to the number of questions that we have come across throughout the world. I think this would be a wonderful platform for people across the world to start their practice into the refractive stages. And let us all help you out the knowledge that we have. And let us discuss some very common queries that you have. And hopefully we'll be able to answer and sort out all your queries. Thank you once again, Kamal, and welcome you all.
Dr. Himanshu, a few words from you, and then you can come up with a beautiful presentation which we have.
Sure. So in fact, rather than wasting time, I will come to that presentation only. That's the opening thing from all of us. In fact, all three of us would share the sentiment. And this is about the phases of learning in life. We all go through such different kind of phases in life in learning about different things in life. The first phase is age of ignorance, where you feel, well, this is not for me. This is useless. Very typically when you don't have a mentor to teach or somebody where you're interested to learn. Of course, all of us have crossed this stage, and that's the reason why you all are here. Then second comes age of innocence. That's where you feel everything is good. You really think that your mentor is the best, most knowledgeable person. You really think this speaker, whomever you are listening to, is the best speaker. You want to follow this person everywhere. And whatever he or she says is gospel truth. The third comes age of arrogance, where you think that I know everything and everybody else whoever is doing whatever, it's totally wrong. It's like age of fault finding mission. I always say that if you only look for dirt, you'll only gather dirt. Then comes the age which is age of acceptance. And I always like to say this sentence that there is nothing right and wrong in science. There may be different ways of doing same thing. And we understand this thing at that time, that point of time. And that time we start accepting people. We start learning from that. You know what? You can learn from everybody. If you want to search for diamond, you have to go into coal. And everybody speaks truth unless proved otherwise, and there's no end to learning. And finally comes the age of Zen, where nobody's opinion matters. What matters is you. You try to improvise on yourself. You keep challenging yourself. And this is what we want to achieve in our life. All of us go through such thing, and hopefully at the end, we would be able to tell ourselves where would we stand and where do we want to stand. Thank you, Kamalbhai, and I'll hand over the proceeding to Sonu.
No, I just need to go through the questions first. Just give me a minute. Okay, now one interesting thing is that we've tried to answer the questions sent from all our friends across the globe, all the countries. So there are a few exceptional questions which are intriguing to us. So we will just demonstrate that on the screen. Now, first of all, as I said, welcome to all friends from India, Japan, Russia, Brazil, Argentina, Spain, Italy, Germany, Egypt, Vietnam, Philippines, Singapore, Malaysia, all the Middle East, and a few countries of Europe. Now, this is an amazing audience which we have. And as of now, I'm seeing we nearly have 1,100 audience, and it's growing. Now, few questions which we would like to discuss here are, the questions would be there. We will, yeah. There are a few questions which are in a language which I don't understand. Fortunately, me or my friends, we don't. Example is Dr. Nicolas has sent some questions in French. I have a few questions in Russian. I'm very sorry, we could not be able to touching that. There are some questions which can best be answered by the company people. Dr. Thomas Jeske has asked, new lens is coming in. Dr. Fernando and Dr. Sujoy Mukherjee has asked a particular question. There are some interesting questions by doctors from India and Philippines and Thailand who are asking me that could be a simpler method, a cheaper method of checking up instrumentation or doing the AC depth, biocompatibility issues. Can there be a piggyback after yak capsulotomy? Now, the most common questions which we saw were a comparison between the ICL and the IPCL, which I'm sure my friend Dr. Himanshu is going to take it up very, very detailed. Sizing questions were approximately 55% of the questions we got on sizing and selection and the optical size. Explantation, we got a lot of questions coming in from Saudi and the Middle East. Early complications from Russia. So these are a few questions. So what we will do is, these questions we will try and include during our talk. And once we finish our talk, we will again see these slides and just kind of review, have we left anything, so that nobody feels left out. And please, friends, do excuse me. There are a lot of names which are not being mentioned here. We are still getting the questions, but we've just made a summary of all the questions which are coming into us. Like Dr. Kuzim Sergey from Russia, this question came to me nearly 45 minutes ago or 1 hour ago. How to fill calculation form for phakic IOL, explantation. There are some Indian doctors, Dr. Sheetal Kishanpuria, Dr. Sridevi. They have specific questions about glares and halos, and what about if the glares and halos are sectorial. So we will try and handle all these questions as we go along. And once we finish, we will answer all the questions. So now up to Dr. Sonu Goyal, and he's going to be talking about his experience with ICL. So we will all switch off our videos and Dr. Sonu Goyal will take over. Dr. Sonu.
Thank you, Kamal. So is my slide display on, Kamal? And my sound
We
audible?
can see the display.
Yes, Tanu, we can see it. Please
We
go
can
ahead.
see. We can see it.
So ICL in my practice, I do not have any financial disclosures in any of the products. So as a refractive surgeon, for me, the spectrum of refractive surgery varies from the cornea-based surgeries, lens-based surgeries, and in extreme immotropes, I combine, and that is what I call as bioptics. So whenever a patient walks to me, I give the patient options, which is available in my basket, in my center. And these vary from as PRK, LASIK, epiLASIK, LASAC, SMILE, conductive keratoplasties, cornea inlays, lens exchanges, ICLs, posterior chamber phakic lenses, clear lens extractions, and then a combination in cases where both can be done together. So the patient is not biased, the patient is given options, and the patient has the right to choose the various options available. A wonderful paper on phakic intraocular lenses, and the various phakic intraocular lenses available in the world market vary from the angle supported, the iris claw lenses, the Veriflex, Artiflex, and the posterior chamber lenses. My talk would be very specific to this subgroup of posterior chamber implantable lenses. So the concept of posterior chamber behind the iris, far from the endothelium. Now, the success of these posterior chamber lenses is because the endothelial cell loss being away from the endothelium, excellent cosmesis because they're invisible when they're implanted. They're very close to the nodal point of the eye. They help in gaining in the retinal size image and the greater effective optical zone because, as we all know, it is almost 1.25 times when it is translated to the corneal plane. So patients with larger pupil can be handled with these implantable lenses. And yes, it is sulcus located. So the stable location allows for the toric design with the same platform. These are easy to remove and it's a reversible procedure. There is no fixation onto the iris. A question was there whether uveitis could be an indication. Uveitis strictly is a no because this can sometimes, this lenses also can evoke some kind of inflammation. So it is better to avoid. It does not alter the shape or remove the tissue of the cornea, so the cornea still remains virgin. So a good outcome is achieved by a combination of a good product, a good preoperative assessment and indication, good surgical technique, and a good postoperative evaluation. The preoperative assessment, patient selection, and the lens calculation, for me, would basically mean a patient in the age group of 21 to 60. The lower age limit can go to as low as 19, but usually in our scenario, these patients are usually with high myopia and usually unstable. So depending upon the stability, which less than 0.5 diopter change in 12 months would give me an idea of the stability of the refraction. So myopia, hyperopia, or astigmatism is what is in the available power range can be addressed with these lenses. The anterior chamber depth, now this is very critical from the endothelium. All my talk, all my words would mean from the endothelium, close to 2.8 millimeters. The angles, grade 3 to grade 4, widely open angles, and no history of previous ophthalmic surgery, especially the cornea refractive surgery. No previous ocular pathology like keratoconus, glaucoma, Fuchs dystrophy, low endothelial cell count, or any other corneal pathology. Now, there are certain off-label indications which we all have been addressing. Patients after refractive surgeries or with refractive surprises, patients after keratoplasties or with DLKPs, patients with ICRs, patients post-cross-linking, patients with stable keratoconus, patients in children's where there is high anisometropia, and pseudophakic, this being used as piggybacking lenses. But these are all off-label indications of all these phakic intraocular implants. There are certain contraindications which are strict no, pre-existing glaucomas, cataract, uveitis, or other ocular pathology that could interfere with the preserving visual acuity. Monocular patient could be considered as a relative, but not an absolute contraindication, narrow angles and small anterior chamber depths. And yes, zonular abnormalities, lens subluxations, and the unstable refraction or an abnormal cornea or an irregular cornea could be one of those indications where these lenses cannot be used. Now, the recommended ocular examination, we all as refractive and cataract surgeons, we all know it is just nothing different from the casual that we do. Avoid contact lenses, yes, because let the cornea get normalized for 7 days. Refraction both manifest and cycloplegic. A good anterior segment evaluation under a slit lamp. Pupil examination is a must in these patients, and a good palpebral aperture evaluation because it's an intraocular procedure. You want a wide opening. Slit lamp examination, including the AC configuration, the iris topography, iridocorneal angle estimation using any of those equipments, the cornea, and the lens evaluation. Now, as far as the measurement goes, the angle estimation, the anterior chamber depth, the corneal thickness, white to white, the keratometry, the axial length, the endothelial cell count, and the intraocular pressures. Now, let me come one by one. The most important and the crucial factor in deciding the fate of these posterior chamber implants is the sizing. And the sizing begins with the refraction and the two key, the anterior chamber depth from the endothelium and the white to white. Now, this ACD can be measured using the pentacam, the IL master, or the OpScan. A lot of questions if you do not have these equipments. White to white can be measured using a very cheap digital caliper, an IL master or an OpScan. Sulcus to sulcus STS, which we call it, was initially being used, was done by the UBM. The angle assessment can easily be done using a gonioscope. And OCT, for the macular examination, because usually these patients can have some kind of a macular pathology which goes undetected. A specular count is must. So sizing basically is crucial depending upon the white to white and the ACD, which are the two critical factors in deciding the type and the model of the lens being used. And this in turn translates into what is called as the vault. And vault we define as the distance between the posterior surface of the ICL and the anterior surface of the human lens. So the assessment of the anterior chamber basically begins with the estimation of the angle and the depth. By the depth, I mean the anterior chamber depth from the endothelium. So we should all check the default settings of the device, properly calibration, and a trained technician who would repeatedly train himself every time that the test is being done. And always check for the contralateral eye discrepancy. If you have discrepancy between the two eyes, you should either switch on to a different equipment or maybe repeat the test on the same equipment. So the central value is only at a single point. We must assess the anterior chamber depth from center to periphery as well as the angles. The iridocorneal angles can be estimated on the slit lamp using a very simple von Herrick grading system that we're all accustomed to. Paper which I just navigated, when you compare the anterior chamber depth on the IL master, the pentacam, and the OpScan, now if you see this paper, which I have highlighted, the OpScan and the pentacam fall in quite alignment with each other, but the IL master tends to overestimate the anterior chamber depth by a figure of almost 0.09 to 0.1. So between the equipments, there could be some variation which has to be taken care. Now, white to white measurements, the simplest and the cheapest method is this digital caliper. And believe me, in my practice, I found it to be the most reliable tool to measure the white to white. So initial 20 cases, I would recommend putting in a paracan drop, take the patient in the OR, make the patient supine, measure under the microscope, read the caliper measure on a calibrated ruler, or use the digital caliper and find out the readings. If you have an access to an OpScan or an IL master, you can easily do that, but be careful in patients with pterygium, patients with pigmented area around the cornea. There could be some discrepancies between the measuring devices and discrepancies between the two eyes. So both eyes should usually have a similar white to white measurements. A close look, if you can have on the white to white on an OpScan, 12.6 is the reading. But if I take care of the eye metrics, look what I figured it out. It has taken beyond the desired, and the actual reading would have been 11.5 centimeter. For 5 millimeters, sorry. So that is what I want to highlight. You have to be very, very careful in the equipment that you're using. So white to white with the IL Master 700, in our experience, it tends to overestimate by 0.3 millimeters. The 500 tends to do it by 0.5 millimeters, and the Pentacam again by 0.3 millimeters. So in my practice, I would always use a digital caliper. And in case of doubt, I would always go back on these equipments, try to correlate between the two and get the most correct reading between the two. The white to white can again be measured by the pentacam or the lens star, and the validation is very important. Again and again, I want to emphasize validation of the equipments is very important. So an ideally sized ICL will provide a vault, as we have discussed, the distance between the anterior surface of the lens and the posterior surface of the ICL within a range of 250 microns to 750. So that is what an ideal vault would be for most of the practitioners. If you do it on a slit lamp, half of the corneal thickness to 1.5 of the corneal thickness is the ideal size that we want. An undersized ICL would stand less than 125 micron vault, and this would increase the risk of the anterior subcapsular opacification. And an oversized ICL where your the thickness goes above 1000 may push the iris forward and cause an angle closure and cause of the rise in the IOP and sometimes beaking of the pupil. Now, this is how once you have taken the clear corneal measurements of the white to white and you've taken the anterior chamber depth, now this transforms into the length, the overall size of the implant which is used. So in patients where you have a white to white, for example, 11.15 to 11.64 with an anterior chamber depth of 3, the lens overall length would be 12.6. So these are the various the manufacturers which have their own nomograms, and once you have the data that you have pushed in to the respective companies, they would come up with the size which they would be delivering to you. The quick word with the endothelial cell count, depending upon the age and the depth of the anterior chamber, these are the basic requirement which is for the endothelial cell count. And a wonderful study by this Japanese group and Kimiya Shimizu himself, who was the designer for the KS Aquaport, came up with the comparison of the corneal endothelial cell density and morphology after the posterior chamber phakic intraocular lens implantation with and without a central hole. And he concluded that there is no difference in the endothelial cell density in ICLs with and without a central hole, except in the superior part, where because of the peripheral iridotomies, there could be some cellular loss. So phakic IOL basically in the world market is available by the Star, which manufactures in the name of ICL. And we have a great company here in India, Intraocular Care, which manufactures by the trade name of IPCL. And 2-3 other companies, Biotech and Apaswamy, other companies are manufacturing, but IOC is one of the biggest manufacturers in the Made in India program. So the ICL from the Star is made up of a collamer material. It is a collagen and hema copolymer, excellent biocompatibility and excellent transmittance. So collamer is basically a collagen copolymer, and the unique property of this collamer minimizes the inflammation, the flare, and the cellular reaction. And this is basically a monolayer formation of the fibronectin, which makes it inert. And on a long term, it makes quite compatible with the human eye. The ICL shifted from the Star from the V4B model to the V4C. Basic difference between the V4B and the V4C is the hole. The V4C has the central hole, which is called the KS Aquaport. The diameter is 360 microns, and this takes away the need for creating a peripheral iridotomy when you're using the model of this particular lens subtype. Then was the evolution in the visual. This is called the EVO Vision ICL, and they came up with an optic diameter in their EVO Plus platform, where to take care of the millennial population with larger pupil diameters, the optic size was increased, and this optic size varied from 5.8 to 6.1. As you see here, with lens size, a lens refractive error of -9, you have an optical diameter of 6.1, which transposes onto the corneal platform at 7.6. So imagine a 7.6 optic zone with the refractive surgery wasn't ever possible with LASIK or SMILE. So that is the biggest advantage. These are available in the toric and the non-toric platform from as low as -0.5 to -18 diopters, and on the hyperopic side from 0.5 to +10 diopters. And the toric design, they are available for 6 diopters of cylinders. The average pupil size between the age group of 21 to 45 years, the mean pupil diameter under night driving conditions varies from 5.4 to 5.2. So that means the 21-year-old guy would have, under night driving conditions, a pupil size of around 5.4, and a 45-year-old guy under night driving conditions would translate to a pupil size of 5.2. So increasing the optic size of these ICLs was one of the measures taken to prevent these patients from getting those halos and glares and possible risk factors for night vision. A lot of patients complain of after the implantation of these glare and halos, but over a period of time, they usually tend to settle down, probably after maybe 1 to 3 months. And this basically boils down to some kind of a neuroadaptation. A JCRS publication here, patients with large pupil diameter could definitely benefit from ICLs or IPCLs with the larger optic zone. And the V5 series, which the STAR people call EVO Plus, and by default, the IPCL by the CARE group has a larger optic diameter. So this is the IPCL from the CARE group, and this has been commercially available since 2014. It was launched, the V2 model was launched in 2017, and worldwide around 60,000 lenses have been implanted. And these are available in the myopia, toric, presbyopic, and the presbyopic toric platforms. So the V2 model has basically a central hole, which is 380 micron or 0.38 millimeter diameter. And the central thickness is uniform in all diopters, and it is around 80 microns. And the water content is 26%. And the most interesting with the IPCL is the range with which it is available, -1 to -30 in steps of 0.5, and +1 to +15 in steps of 0.5 on the hyperopic side. And for the cylindrical correction, they offer to almost +10 diopters in steps of 0.5. And for the presbyopic side, they give you addition of 0.5 to +4 in steps of 0.5. And they're quite compatible, going through incision size of 2.2 and 2.8. There is a wonderful design here with the central hole, where posteriorly the size has increased. They have increased the dia to 420, and anteriorly it remains 380. So the basic idea is to provide a slant, and this slant basically translates into less loss of light as comparison to the vertical, where the anterior and the posterior diameter of the hole remains equivalent. So this basically translates into some kind of a lesser glare or halo phenomena as well. One beautiful interesting of this is that its tolerance to 0.5 millimeter of the white-to-white error. These are the specially engineered haptic pads with the spring effect. So even if you have a slight miscalculation, this is quite sparing, and the vault almost remains stable for almost 0.5 millimeter of the white-to-white errors. This is from the biotech, and these are the specifications, and this is a hydrophilic, and this is the alignment of the third platform available. Recent study published in the IGO, where Dr. Gitan Shah et al. Came up comparing the clinical outcomes of the posterior chamber phakic intraocular lenses, and they concluded that the results of the ICL and the IPCL in terms of safety, efficacy, and predictability and stability was similar. IPCL is an economical viable option for the treatment of myopia, myopic astigmatism worldwide. Now, this is how the sheet, once you have taken care of the diffraction, the anterior chamber depth, the keratometry readings, the intraocular pressure, and the white-to-white, the data is being sent to the respective companies, and these companies will pull you this kind of a calculation sheet, which will give you the model of the lens to be used, and in case of a toric lens, the alignment of the toric axis. So intraoperatively, you need to take care of marking the cornea, the pre-op preparation, loading of the lens, the surgical technique. A quick word of the toric. We all are cataract surgeons, and we are all used to using toric lenses. So what I do is I take the patient on the slit lamp, keep the head posture straight, put in a drop of Paracane, and then mark the 0 to 180 by rotating the slit lamp beam to 180 degrees. And then you can just put in stain, or you can just scratch with a 26 gauge needle. And that is the most accurate way to give you the reference marking, 0 to 180 degrees. And this is intraoperatively. If you don't have an access to an electronic marking system, you can simply use the Mendes device, which is at arm. These horizontal markings are the alignment axis, which are translated onto the lens, and these can be then aligned with the reference axis. The surgical technique I will take you through. Size, size, and then gradually pushing in the lens. Mind me, the trick is the lens, like in cataract, has not to be pushed aiming posteriorly, but this has to be directed towards the angle. And then gradually the manipulators just need to hand through the blades, never touch the optic across on the opposite side. Push away the viscoelastic and then gently so a no touch technique. The optic is the most delicate part. This is the merosul stone. Very carefully, you can draw the ICL. This is the specialized cartridge system. A drop of viscoelastic. Now look at this specialized sponge. I'm trying to nudge the lens to the cartridge. Not using any forceps. The optic is finished. Should not be crushed. What is important? You have to know the holes. The middle and the lower are in line. Now, mindfully pushing the guide which will help you preventing upside down. Specialized forceps. Go through the side, grasp the ICL, and you can just withdraw the cartridge from the left hand rather than moving the right hand. And this is the loading of the lens. This is the specialized center. The soft sponge pushed to the side. And you hear a click. The moment there is a click, position. It should be well hydrated, because if it's underhydrated, there are chances that the lens can get entangled. Push this in. Push in a drop of viscoelastic and then lock it. And the delivery system is ready. You just need to grasp properly and then scoop in a very controlled manner. The input size 2.8 to 3. This is 1 millimeter clear format temporal. 90 degrees apart, 180 degrees apart, and a clear cornea, 3 millimeter incision. I take all my patients under topical anesthesia. HPMC or Delon is your personal choice. I always use HPMC. Two advantages: the chances of rise of intraocular pressure is less, and certain studies which say that there could be I use my left hand to support the jaw. Very slowly push the and just carefully mark the holes in the center. I would always look at this hole and this hole, and if they are going in line each other, you will never have the lens going upside down. Once you reach this stage, you push in some amount of viscoelastic in the center and gradually nudge. I'm holding this junction here. I'm not crossing here, and Similarly, this goes, and this is the plate and the lens junction. Gradually nudge it under the iris. I go through the main room now, and this junction, and gradually nudge again, main room, and nudge, and then let the lens dial the desired axis. This is the workup sheet that has been supplied by the respective manufacturers. Access to amenities. In fact, I have an access video telescope. I can align the lens. A good viscoelastic wash is essential, whether you are using it or want that it comes in a bolus, and viscoelastic has to be removed completely. Just watch. Point which I want to emphasize here again, this hole, both are moving in line to each other. This is a toric, so you can again see the alignment axis, and once they are in line to each other, you will never have a lens moving upside down. Viscoelastic goes in, then gradually you can nudge the lenses underneath. Viscoelastic wash, very important. And the wound can be what if the lens tends to go upside down? Very initial cases, areas, and now watch carefully. The lens tries to flip. I'm trying to push, going in, and look, it goes all the way upside down. The lens is very soft. You can use the same lens grabbing forceps. Push in the viscoelastic, just hold the plate, just draw it. It comes out the same, and you can reload the lens. So these are the special ICL positioning instruments, any axis which you can use. I've been using the manipulators, the Vukes ICL manipulators, and they work wonderfully. Post-op medication is the surgeon's standard procedure and medical judgment. Combination of antibiotic steroid. I usually give it for 2 weeks, a very soft steroid with an antibiotic. Now, what do I look for post-operatively? For me, the most important is the vault, and a low vault in my fear could lead to a cataract. A high vault could give me secondary glaucoma. I would always, in all follow-up visits, do a specular count. I would always do an eye tracy to check the alignment, or maybe a simple refraction, which will give me an alignment. And under dilatation, I can do examination on the slit lamp, which I can check the toric alignment and the checkup of the intraocular pressure. So this is my routine protocol for the checkup and the visits. Day 1, 1 week, 1 month, 3 months, and then 1 year. So post-op care, early 2 to 6 hours, you need to check in on the slit lamp that the wounds are sealed, there is no leakage, the AC is formed. You have to look in for any abnormal cells, flare, pigmentation, the IOP, and the ICL vault, and you have to look in for the centration and the orientation if it's a toric lens, and you have to check the patient having medication properly or not. If the IOP is high on day 1, the most common cause is the retained viscoelastic. There could be insufficient PI. If you're using a lens without a central hole, the PIs could be blocked, or you are somewhere with an oversized ICL. A study done by a group of Salem Al-Maki et al. The post-op day 1, the most common cause of rise of intraocular pressure in the group came out to be retained viscoelastic. And between the 2 to 4 weeks, it was the steroid responsiveness. High vault and pupillary block attributes to almost 10% of the pressure rises. So post-op care continues for a month, and with the uncorrected visual acuity, the biomicroscopy, including the anterior chamber angles, the ICL vault, recording of the pressures, and Even a slit lamp examination gives you a rough estimation of the vault, depending upon comparison with the corneal thickness. A quick table which gives you comparison when it is almost one thickness of the cornea. That means it's almost 100%, and it closely gives you a vault of 550 microns. So objective quantification of the vault can be done using either a anterior segment OCT or a pentacam or a UBM. And this is how the picture looks. You can see the gap between the human phakic lens and the posterior surface of the ICL. And this is how the vault looks like. Dilatation examination, where you can have a look of the alignment of the toric ICL. So the anterior segment optical coherence tomography, angle and vault analysis after toric and non-toric implantable lenses, there is definitely a significant reduction in the iridocorneal angle after the ICL implantation. And a comparison study of the three anterior segment tools used to measure the vault after the ICL implantation. So the anterior segment OCT demonstrated a little higher value of the vault as compared to a pentacam or a UBM, but still all the three equipments can be used when it comes to vault measurements. A wonderful paper comparing a vault after implantation of both a lens with and without hole. There was no significant difference. Again, it was this Japanese group which came up, and the vault remained equally in both the subgroups. We'll be talking about the lens opacities. Before that, I'll just quickly take you through the troubleshooting which I've compiled. So post-operatively, if you have a patient with a high vault, but the pressures are normal, you need to observe such a patient. But if in case you have high intraocular pressure, the chambers are shallow, you can rotate the lens vertically if the lens is a non-toric. So by this, I mean, if you have placed the lens 180 degrees and the chambers are shallow, you can just go in the anterior chamber, rotate the lens vertically, and yes, it will settle down. The chambers will definitely become deep and the vault would tend to decrease because the internal depth width horizontally is lesser than vertical. So it can compensate. If you have a toric lens, you need to exchange one size lower. If you have a patient with a low vault, but there is no peripheral touch, but you have a lens which has a central hole, for example, the V4C model or the IPCL with a central hole, you can keep the patient under observation. Still, you have a patient with a low vault, but there is a peripheral touch and there is no cataract, but it's a central hole. You can still observe the patient. If you have a patient with a low vault, a peripheral touch, and there is an early, but that's a non-progressive opacity, you can still observe in cases of a V4C model. But if you have a cataract which is progressive, you need to explant or you need to exchange with one size up if the patient is insisting for a refractive solution. In case you have a toric which has rotated, you need to go in a lens with one size up. This paper done by George Alio gave the explanation to the causes of explantation in cases of phakic intraocular lenses. And in the posterior chamber group, it was basically 65% of the patients were explanted ICL because of the cataract. So the biggest cause of explantation in this PCIL group is basically cataract, and little can be attributed to the endothelial cell loss or the corneal decompensation or the lens dislocation or intraocular pressure rise or ovalization of the pupil. Some patients have retinal detachments, and one patient was explanted because of the glare and halos, which was persistent in these patients. So quick review of the potential complication. Cataract was the major postoperative complication reported in 136 patients out of 2,500 eyes done. And the most common cause of cataract still remains the intraoperative manipulation, and it is this which is the major cause. So thank you, and I like to conclude, and I like to hand over to Kamal. And
very nice, Sonu. It was such an exhaustive and comprehensive overview about entire gamut of phakic are beautiful, beautiful.
I think you covered practically everything that makes mine and Himanshu's job a lot
Yeah,
easier.
we can go home, might as well.
Yeah, but I guess what we'd like to just bring about here is 2, 3 points, which
Yes.
came out of the whole thing is before we start on to Himanshu's discussion was the incidence of cataract after the central hole has been introduced is phenomenally gone down. I guess there are a lot of studies which say that the stasis of the aqueous behind the phakic lens was probably one of the more reasons than the actual contact between the phakic lens and the anterior surface of the crystalline lens. So with the hole being central hole being there, I think this thing has gone down tremendously. Further, there is a lot of hue and cry about cataract being induced by the lens. Well, I would say my personal opinion, I've nearly done more than 2,200 lenses till now. I have just got cases who have got cataract which were due to a bad calculation, not the lens by itself. It was a faulty calculation. It was during the learning phase, or these patients got some trauma, or they had a retina detachment and there was a silicone oil and the cataract. So by itself, I guess cataract is much more overhyped than we actually see in the market, which I'm sure all three of us would agree. And another interesting thing is the sizing, which I guess I'll be touching a few salient typical situations in the practice. Dr. Sonu's wonderfully covered it. Now I hand the stage to Dr. Himanshu again. And again, I think he's going to take us through an amazing journey about how to choose a phakic lens and decide its sizing. Over to you, Dr. Himanshu.
Thank you, Kamal, and very good evening to everybody. So my job partly has become a little simpler because Sonu has taken care of the basic part of it, and I might pick up a few things from everywhere. Before I start anything, let me have a clear disclosure that none of us have any financial disclosure here. I'm not a consultant to any of the company, any of the product which I'm talking about, and certainly not biased or forced by anybody else's opinion. Having said that, everybody is entitled to an opinion, and so am I. So I'll go ahead with something which I believe, and I'll share the journey along with my understanding, which I'll share with you. So two major phakic oil, as Sonu said, that there are currently in Indian market, at least there are 4 or 5 phakic oil, but world over, there are two major companies which are in the forefront of sulcus-based phakic oil, and that's from the Star Vision ICL and from the IOCare IPCL. So I'll be sticking to these two, and there are a lot of questions which came about the comparison. So I'll be taking one-on-one on each different component of that. Please let me clarify one thing in the advance, that my journey started with ICL long back, almost 14 years back. And I use both the lenses, so I don't have any love and hate with any of those things. So the interesting part of this entire topic started with a David Goliath kind of story, where there was one company, big, huge multinational company, which came and swept the market and moved away those iris-based phakic oil, and they didn't have any competition. And there came somebody who was a smaller company and who took them by horns and came with their product, which was IPCL. So if I compare both of them, Sonu rightly showed that it's a implantable lens, and it's available in the wide range ICL, which is from 0.5 to 18 in spherical power, and in cylindrical power also almost the same range. It's pretty simple surgery. Let me tell you, if you are able to do any intraocular surgery, this surgery is as simple as any surgery. If you're doing cataract, well, believe me, you're doing much more complicated surgery than this surgery. So it's very simple learning curve, but you must understand that there are certain nuances which we have to understand. As in, the lens sits into a ciliary sulcus, which is not visible to us. And whatever measurement we take are surrogate measurements. So sometimes there can be a mismatch of the sizing, and that leads to the problem. We'll come to that. The second lens I'll be touching up is the IPCL from Care Group. Very similar design, again, plate haptic design, and we'll take each thing one by one. Now, earlier when it came, in fact, I was also reserved that why should we change over to anything? And I resisted a lot and started using these lenses just 5 to 7 years back. Currently, there are more than 60,000 lenses implanted across the world, more than 1,000 surgeons in India, and more than 2,000 surgeons across the world. So it's certainly not a new lens. Whether it works in your hand or not is something which we all have to find out and fine tune why not. So let's start with the first thing, material. A lot of you ask us that what is this thing about comparison between material of these two lenses? And hence I'm starting with the material part. Now where Star came and swept the market was the Collamer part, where it says it's Collamer, which is basically a combination of telecollagen from porcine origin, along with polymer, which was hydrophilic and hydrophobic acrylic combination. Now, there's a story behind this thing. We all heard the story from our company, biocompatible, biocompatible. But interestingly, when you dig down a little bit, you find out that the patent about this thing came long back. In 1994, first patent was filed for the Collamer. Now, this Collamer material was basically used, and you can just search this thing on Google patent. It's available, this document. The reason behind Collamer that time, 1994, there were no good hydrophilic IOLs available that time. There was beginning of possible the era of Bausch & Lomb Acryos lens, where the hydrophilic platforms or centroflex lens from Rayner, these were the possible beginning that time. And that's the time where they were trying to find out a molecule which can work as a hydrophilic polymer. The problem with hydrophilic polymer was that when you use hydrophobic, there was a protein deposition on that. Hydrophilic material used to swell beyond limits. So when you put both of them together, the lens inside would swell up uncontrollably. So they introduced one coating, almost 0.2% coating by telecollagen from porcine origin, and the swelling of the lens reduced, and it rather stopped. And there, voila, we had a Collamer. So the entire idea of getting into Collamer was just to reduce the hydrophilic lens to swell. It was nothing to do with any biocompatibility or something. Of course, it was biocompatible. It was, in fact, used first in intraocular lenses. But later on, they realized that, well, we can use it somewhere else also. But till then, the hydrophilic lens technology evolved. There are so many hydrophilic lenses came into the market, which was stable, which did not have any swelling, irrespective whether they use Collamer or not. So the Collamer part, which we love to love, is partly related to the industry, which is trying to convince us that Collamer is the most important component of the lens. Unfortunately, no. Hydrophilic component is equally good. Now, Sonu took us through this thing that they both are water-based lenses. They are hydrophilic lenses. ICL has a little more water content and IPCL has a little less water content. Does not matter. All these material are very stable and they are extremely biocompatible material. They do not lead to any cataractogenesis changes, which we have studied long back. So that's about the material. Then comes the economic part of it. Now, why should I use one particular lens, or why should I use phakic lens? Well, somebody asked that what is the basic setup you required, in the question somebody has asked, that basic setup required to start a phakic IOL. And nothing. If you are doing an intraocular surgery, your investment is almost nothing. All you need is just a digital caliper, which is just a normal vernier caliper, which is available widely everywhere. And you need just lens adjustment instrument, which Sonu showed, and which are again quite cheaply available. So you do not need any great kind of financial investment to start phakic eye surgeries. When we compare these two head-to-head, Vision ICL and IPCL, well, IPCL, because we are dealing with multiple countries here, I won't be able to give you exact figure in your country, but we all know if we compare, almost 20 to 25% IPCL cost-wise comes less than ICL, which is a good thing to remember, because that reduces the burden on surgeon as well as the patient. Then comes, let's compare the power of each other. The power part, when we look at the ICL is available from 0.5 to 18 diopter myopia, whereas IPCL by default is available from 0.5 to 30 diopter, which is a huge amount. From 19 to 30 diopter and beyond. And Kamal will show you the videos where he has used lens beyond 30 diopter also. So such high myopic lenses are also possible. So these lenses, IPCLs, are customizable. Does it mean ICL cannot be customized? Certainly not. But the logistic related to the customization of ICL is very difficult, because it's a very big company, centralized production. It may be a little difficult for you to get a customized lens from there, and cost-wise also may not actually help us a lot. Next comes the size of the lens. When the lens size are measured in millimeter, and the ICL is available in 4 sizes, from 12.1 to 13.7, whereas IPCL, which is in the steps of 0.5, whereas IPCL is available in 13 sizes, where it is available from 11 millimeter to 14 millimeter in the steps of 0.25, which is quite amazing. And again, it's beyond that, it's customizable. Somebody wants 10.5, I'm sure they can manufacture that. Somebody wants 15 millimeter, they can manufacture that. That's customizable. The trick behind here, what we have to understand is the strength of the IPCL can sometimes become a problem with IPCL, because it's available in the steps of 0.25. You may end up taking a lens which is possibly 0.25 millimeters smaller than the ideal size, and you may end up getting a smaller vault, which in ICL may not happen because you do not have a choice. You just take steps of 0.5. But I'll come to that later on in the sizing part, which will tell us that how we can avoid such kind of thing. Next comes the design part, where the hole came. Now the hole was introduced in ICL first, which showed us that before that, the lens, we needed to do a peripheral iridectomy, whether intraoperatively or laser-based, but we had to do that. But when the hole came, it made the procedure one step, as well as it didn't require any more invasive procedures like peripheral iridectomy. The hole, Sonu nicely correlated both the holes, and what IPCL claims that their hole is slanting and hence gives less glare. Now, is there any clinical evidence to that? I really don't think I can say yes or no to it. Does it do anything bad? Certainly not. Now, to study the glare, and some people have asked about the reason behind glare and those things. There are multiple reasons from the material to the edge of the hole, which can lead to glare problem. Now, interesting thing is it's very difficult to study glare problem clinically when you are dealing with any instrument which we have in current science. Because whatever we do, we measure all the aberration and everything end on. We measure, we throw the light straight, we catch the light straight back, and then we talk about the aberrations there. But we do not have any way where we take the light from the side and then take the aberration which are coming and something. So in real life, if somebody is driving and they get light coming from the side, they can have glare, and which is very difficult to study clinically.
Camera off.
Right? So that's something which we have to know about. Now, the hole what we have is to allow the aqueous to pass through so that there is no pressure buildup behind the lens, which can lead to increase in intraocular pressure. Remember, it's for the aqueous to pass through. However, there are certain other holes which are there in IPCL, which are holes on the optic, where you can see a couple of holes on the optic junction, which allows the fluid to pass through that. Now, what's the advantage there? Well, it allows more fluid to pass. So potentially there can be less chances of cataract because there's more aqueous flow. Is there a clinical evidence? We really don't know. We have to wait and watch. But what can be the disadvantage? Where disadvantage can be glare. But interestingly, these two holes are placed superiorly, which is covered by your lid, and hence it is actually fine. It's covered by pupil, hence most of the time we are fine. So there are certain myths about these holes which are prevalent. So when we started with the hole, we said, you know what, now no glaucoma, we are absolutely fine. But I told you that this hole is meant for aqueous and not visco. So you may not require to do a PI intraoperatively for aqueous to pass through. But if there is visco there, it's not going to pass through this small hole. It may clog the hole. So now a bigger PI, which was allowing the visco to pass through, the smaller hole may not allow the visco to pass through. Hence, we have to do better visco wash than where we used to do with PI. So remember, the IOP can be high if you're not done enough visco wash. There are other reasons of glaucoma like size mismatch, pigment dispersion, chronic inflammation, steroid response, which will still hold true. So hole does not mean it will not lead to any raise in intraocular pressure. The other part of the design which we have to compare is optical diameter. The previous lens which was available in Vision ICL was a little smaller lens, 5.1 to 5.6 millimeter. Again, the lens diameter changes with power. Please remember, no lenses, whether ICL, IPCL, would be same in a low diopter versus high diopter. So the diameter keeps changing. Typically, the diameter is maximum with the power, which is intermediate power, like say between 4 diopter to 10 diopters. There you get the maximum diameter. So now the new EVO Vision IPCL and EVO Plus gives you 6.1 millimeter diameter, larger optic, whereas the version 2 of IPCL gives you up to 6.6, pretty larger optic than that also. Having said that, this reflects to almost 7.5 millimeter diameter or 7.8 millimeter diameter at the corneal plane, which is huge. Most of our pupil are not that. Especially somebody has wider pupil, please note down, because you can customize some lenses like IPCL. Again, Kamal would talk about those things. Then comes the loading part. Now I'm comparing loading between ICL and IPCL. The loading for ICL is done from the front loading, where you pull the lens into the cartridge, where you need a special lens holding forceps. You hold the lens and then pull the sleeves or the cartridge away and set it inside. Whereas the lens like IPCL is backloaded. It's like a normal butterfly cartridge of a foldable IOL, where you put the lens and load it, which is pretty simple, and you don't need any special instrument. But there's one thing which we have to remember. One unique thing between these two is the loading part. After loading, the ICL is convex upwards, whereas IPCL has to be concave upwards. So when you are folding, you press on the center part of the lens to keep it concavity upwards, which prevents any untoward incidents like flipping the lens or opening on the wrong side, which we have to remember. Then comes the implantation part. Though both of them are pretty similar in implantation, but as Sonu showed, that if it's a toric lens like toric ICL, we have to first mark the horizontal axis, and then the company would provide you with a chart. According to chart, you have to rotate. Now, as You can see my patient's chart. It tells me to rotate counterclockwise 20 degree. And that's the lens which is available, which I have to implant. Now, does it mean it's something to do with the patient? No, it's something to do with the availability of that axis and that combination of sphere and cylindrical power with the company. So that's the reason they told you to rotate this thing. If they had an available lens which was similar to your size and your refractive error, you would not have required to rotate this thing. And their IPCL actually stole the show. Well, what they did was they said, you know what, we will customize every lens as per your order. You send us the order, we will make a sphere and cylinder according to that. In fact, not only that, when we send the order, we send sphere, cylinder, size of lens, pupil size, whatever you want. Based on that, they personalize the lens. So it's not even customization. For each individual patient, it's personalized. One question was there, how are the manufacturing process involved here? Well, the manufacturing process for both of them, it requires lathe, and it's basically lathe cutting using CNC machine. Right. Now, CNC machine are computerized machine. There's no human involvement in the lathing part, and the error involved there is just 5 micron, right? So please rest assured, if the entry is proper, which most of the time now is very automatized, there's no chance of error. But whenever you measure the lens, you always measure the lens diagonally, right? So the lens, when we see, is measured in a diagonal position like this direction. So this is the diameter of the lens. Please do not measure in long axis. That's not the diameter of the lens. Diameter of the lens is diagonal, right? As you can see my cursor moving. And so all the lenses, whether it's ICL, IPCL, the measurement is always diagonal, right? Now, as I told you, it became personalized. Now for us now, if I'm using a IPCL or ICL, I mark the horizontal axis. I like to take patient in undilated pupil condition. And one most important part, what I prefer to do here is swing my slit lamp both the side. So when my beam is horizontal, it is passing through this eye's pupil. When I swing it on the other side, if there is no head tilt, it has to pass through opposite side pupil also. If it does not, if it is passing a little bit above or below, that means there is a head tilt. And I adjust the head of the patient, and according to that, I mark my horizontal axis. After marking the horizontal axis intraoperatively, well, you can use either saline injection like hydro implantation. What I'm showing here, I'm just using BSS to load the lens. As I loaded the lens on the front part of the injector, and then I'm marking my horizontal 0 to 180 degree axis. The hydro implantation is very simple, and probably since last 4 or 5 years, I have resorted to hydro implantation. And in fact, I've done a randomized control trial between hydro and visco implantation, and they both are pretty good, equally good. So after making an incision, which is 2.8 here, I put my irrigation port and making sure that my lens is injected a little slowly. As my lens goes inside, I just tip my irrigating port above the lens so that it does not jump up. And then with the lens adjustment instrument, I tuck the lens. Now here, what I'm doing is I'm rotating the lens while tucking, which is very important step. When you tuck the lens, you always rotate clockwise and counterclockwise, which opens up your pads, which are closed down or kinked. They open up and they open in a position. From there, the lens would remain normal. The trick here in hydro implantation is to keep your bottle height at 50 centimeter of water from your eye level, which is very important. If your bottle height is very high, you will have an iris prolapse. So keep the bottle height low. That's the trick there. Once you implant the lens and keep it horizontally, that's the end of the surgery. Now, another thing which IPCL came was the phakic, presbyopic phakic IOL, which was one of its own innovation, where It could be used in lenses or the cases where there was just presbyopia and not much significant refractive error also. Even it can be used along with the refractive error, but this was something which was a path breaking on its own. So if we compare David versus Goliath story here, IPCL gave really good fight to ICL in every aspect. So if I compare both the lenses, I would say both the lenses are good. Whatever suits you, please go ahead. None of them is a bad lens. If it works in your hand, it's absolutely fine. But if you are stuck to one lens and suddenly you change to another lens without understanding the basic of the sizing, you may end up in trouble and you'll start blaming the lens. It's not the lens, it's the sizing part which matters. So both the lenses are pretty same, still they're a little different. And this brings us to the sizing part of my talk. Now, the sizing part mostly Sonu has taken, but I'm going to give you a lot of small clues and small interesting stuff about sizing. The vault we know is the gap between the back surface of the phakic IOL and the front surface of the lens. And that's where we call vault. Vault is usually measured on the anterior segment OCT, and that's where it's perfect and that's beautiful and best way to measure it. The ideal vault is 250 micron to 750 micron, but again, don't go by numbers. It can range sometime from 90 micron to 1000 micron. You still can have some variation there and you still may be fine. Now, what happens if the vault is high? If the vault is very high, as you can see here, there can be rubbing of the iris behind the lens, optic and haptic, which can lead to pigment dispersion and which can actually lead to open angle glaucoma and which can be quite disastrous. If the vault is high, it can actually lead to distortion of the pupil. It may not constrict properly. The pupil may be distorted. Pupil may lead to a poor reaction to the light. And that's what means where the vault is actually very high. The vault is very low. It can lead to early cataractogenesis. Or if it is very small, well, it can lead to decentration. Or if it's toric, it can lead to rotational instability. Please remember that. Now, when do we decide vault is reliable? It depends on what method you use. If you use hydro insertion, like what I showed you video, well, you can directly take a cue whether it's reliable or, I mean, it's reliable immediately. But if you use visco, if the visco is in front of your lens, it may show you falsely low vault. If visco is remaining behind your lens, it may show falsely high vault. So we have to wait till visco clears off, which may take 3-4 days time. So do not comment upon vault completely on your day one. Also at the long run, the vault may change. In fact, vault can reduce with the time in most of the myopic patient as well as even the other patients. Coming to what are the things which we need, parameters which we need for the ordering. Sonu said that refraction, keratometry, AC depth, and all those things. But the most two important thing which we are looking at here is AC depth and horizontal wide to wide. The refraction has to be taken into individualized, especially when you are dealing with uniocular condition. Please make sure you do a contact lens trial. Let me give you one example of my patient. Uniocular high myopia. I did a ICL and I was really happy because the outcome was amazing. Patient got unaided 6/9 vision, which is 20/30 vision. AC depth was very good and I was really happy. But patient was extremely unhappy because patient had binocular diplopia. Patient happened to have small hypotropia or vertical squint of 7 to 8 prism diopter, which patient was not able to fuse. Unfortunately, I had not done a contact lens trial prior to this thing and had to explant the lens. So whenever you have uniocular anisometropia, please do a contact lens trial and look for diplopia. Whatever we do, irrespective whether you use ICL, IPCL, you are going to come across condition where you have to expand the lens. You are going to come across the lens where there's going to be size mismatch, and there the sizing comes into picture. AC depth is measured by multiple instrument, which previous speaker took apart. But the important thing to remember is that the cutoff values are 2.8 for IPCL and 3 for ICL. But does it mean 3 millimeter is totally safe? Certainly not. Whenever you look at such kind of values, you are looking at the central values. The periphery of the iris may be convex, and your central anterior chamber depth may be fine, but the periphery may be actually a problem. Sometimes you can have something called crystalline lens rise, or CLR, which is very high, which is where you join the 2 points of the lens which is touching the iris, and see the lens above that, which is crystalline lens rise. If it is high, you may end up in very low vault. So be careful about such kind of condition. Always look at the configuration of iris on your pentacam or your ASOCT. Compared to that, white to white is equally confusing. The white to white is best measured with the digital caliper. But remember, digital caliper or whatever you take, your measurement is between the zone of clear cornea to the peripheral gray zone or blue zone. It's in between that. And you take your digital caliper, and ideally you should measure on surgical microscope. But most of the time we all use slit lamp with the diffuse illumination. What we try to do is we simulate the surgical microscope here. But remember, it's very subjective. In my hand, I would say there is huge surgical, huge bias here. Now, if you look at the different methods of white to white measurement, this is my own eye, my right eye, and I have measured my white to white on different instrument. Pentacam, it gives 11.8, IOL Master gives 12.1, Keratograph 12.3, eye design 12.3, eye trace 12.1. There's huge variation. What do I choose? But the best thing is, what do I choose is, I take two methods. One method as my baseline, which in my hand, I take a manual measurement on the pentacam where I do image inversion and I take measurement. That measurement I keep on my digital caliper. And now I go in patient's eye and measure and check whether this is correct or not. If not, I'll adjust there and then I decide what would be my size. Ideal thing to understand would be, well, we can measure sulcus to sulcus. That's the best thing. And in fact, there are certain machine which gives you simulation also. Does it mean sulcus to sulcus is the best thing to measure? Well, unfortunately, no. Whatever measurement we take, sulcus to sulcus versus white to white does not have very good correlation. In fact, whenever we look at that, we ideally assume that there is sulcus to sulcus horizontally, which is 0.5 millimeter larger than white to white, but that's not actually true. The correlation between sulcus to sulcus and white to white is extremely poor. Hence, whatever method you use of white to white, there are chances that you may end up in size mismatch. Also, the mismatch between white to white and sulcus to sulcus depends upon the high myopia or low myopia. Higher the myopia, more the mismatch. And phakic IOL are reserved for high myopia most of the time. So most of the time we are possibly going to have mismatch. Now, does it mean STS or sulcus to sulcus is best? Unfortunately, no. In fact, there is a lot of repeatability issue with sulcus to sulcus measurement also. So even with the sulcus to sulcus measurement, there is going to be a variation. And remember, no company is going to ever tell you to switch over to sulcus measurement. So remember, even if you want to do sulcus to sulcus, always keep your white to white in your hand, correlate both of them. If there is significant difference, go for multiple area measurement of your sulcus to sulcus and white to white, discuss with the company and then decide. Do not directly jump over for the order. the final part of my presentation, I'm going to take you through very nice, tricky situation, or rather my tricks, which I use for the sizing of the IPCL. Before I tell you that, well, simple thing. A fixed size lens, when you put in a wide to wide, which is larger, will have less vault. And when you put a lens in a smaller wide to wide, will have larger vault. Everybody with me, right? Now, remember, according to the calculators, these are the possible calculation which you can get. So let's take an example. Left side is ICL. The right side is IPCL measurement area. Now, if your wide to wide of the patient is 11.7, according to the ICL calculator, it will tell you to take the lens of 13.2 millimeter, which is actually 1.5 millimeter larger than your wide to wide. If your wide to wide is 12.3 millimeter, still computer will tell you, or calculator will tell you to take 13.2 millimeter ICL, which is just 0.9 millimeter larger than your wide to wide. So where do you think vault will be high? Well, of course, the lens with a wider difference or more difference. So whenever you are dealing with wide to wide, which comes in the range, which is the lower part of your range of that particular size, and where your anterior chamber depth is low, you are going to potentially have problems. So if you are dealing with a deep AC, you're fine. But if you're dealing with a marginal anterior chamber depth and your wide to wide is coming on the lower part of the range of that particular size, you are expecting problem. Same thing holds true for the IPCL, right? So if you are dealing with such kind of thing, let's take an example of toric IPCL. Now, toricity. Now you have a wide to wide with 11.6 millimeter or 11.65. According to calculator, it will tell you to take 13 millimeter IPCL, which is 1.35 millimeter larger. If you have 11.93 millimeter, it will still tell you to take 13 millimeter, which is just 1 millimeter larger than the wide to wide. Where do you think the vault will be low? Well, the second part, vault will be low, where the difference is less. Now, low vault means there can be potential chance of rotation. So if I'm dealing with a phakic IOL where I'm needing toricity there or toric IOL, rather than my calculator telling me to use 13 millimeter lens, I would go ahead and take 13.25 millimeter lens, which is 0.25 millimeter larger than what my calculator told me. Now, what does it mean? If I take larger 0.25 lens, my vault will increase. But what if it becomes too much? Interestingly, whenever you increase the size, the vault increases by 40% only. That means if I increase the size by 0.25 millimeter, my vault will increase by 100 micron only, which is fine. So when you're talking about toric stability, I would rather take this size compared to the previous size, what my calculator told me. So please remember to get used to what your case scenario is rather than what your calculator tells you. Lastly, I would go into the explantation scenarios. When do you explant the lens? Well, the lens which is damaged intraoperatively, you explant. Lens which is flipped, you explant. Very high vault, where you recognize intraoperatively, you explant. Very small lens which is rotating intraoperatively, you explant. Early post-op, if the vault is too high, you explant, and especially if there is pupil which is not dilating, it's not reacting. Asymmetric vault, when you have one area which is actually high, one area low, you can rotate the lens and see if it stays. Vault is too low, less than 90 micron or lenticular touch, you explant. If there is a rotation of the lens which is more than 15 to 20 degree, most likely you need to explant. But if it is less than 20 degree, it can be related to folding of the haptics. You can rotate the lens clockwise and counterclockwise, and it may do the trick. Late post-op, if it is cataract, severe glare, pigment dispersion, very high refractive change beyond LASIK improvement, uniocular diplopia, you must explant. So before you decide about using or not using any lens, please remember proof of the pudding is by eating it. As Gandhiji rightly said, first they ignore you, then they laugh at you, then they fight you, and then you win. Thank you very much.
I think that was one of the most exhaustive sessions the audience could get a treat to on phakic lenses, and how beautifully both the previous speakers have wrapped up the whole thing and set the stage for me to walk in. I think most of the points, questions which came to us, I think we got a huge number of questions, and my fellow speakers agree with me. We had to stop looking at the questions till 3 p.m. in the afternoon because they were coming like crazy. So we had to stop them. But one thing I'm going to do is, since we have access to the emails, we'll be answering back to the questions, and if possible, directing to the particular speaker. Number two, I think the success of this talk is amazing. We will continue with this Eye to Eye series on different topics with fantastic speakers all over. I think what points Dr. Himanshu has brought about are very, very relevant. And they not only just help a beginner surgeon, I think even a prolific surgeon would take on some gold points. So I guess most of the audiences are good surgeons, as Dr. Himanshu said. Any average phacosurgeon can be a great phakic IOL surgeon. The only thing you need to keep in mind is when you're doing a cataract surgery, you're working between 5 to 8 millimeters, and now you're working between 2 to 3.2 millimeters. Number one. Number two, the sacrificial lamp is the anterior capsule in your cataract. Here, it's a very pristine, precious tissue. So you need to be watching out for that. So we all are trained to look at the endothelium, protect the endothelium as cataract surgeons. So our all focus is looking at the endothelium as a cataract surgeon. Well, here comes the new punch. When you become a phakic surgeon, you have another baby. The first baby is your endothelium. The second baby is your anterior capsule. So we need to be careful how we look at that. Another thing, I'm just for the sake of repetition, the way Himanshu beautifully explained, the way an ICL is loaded and the way the IPCL is loaded. The difference is very, very important, because if you load the IPCL the way ICL is loaded, you will have the IPCL going and touching and rubbing the crystalline capsule, and you do the reverse with an IPCL, it will not land behind the place you want. So very beautifully elucidated. I think again, few points which Himanshu said was sizing. Nearly 45% of questions we got were on sizing. I'm sure once this is over, all sizing issues are gone forever. Now, what I'll be bringing forth would be fine tunings in sizing, where to use your own mind, where to add extra information. Suppose you're going in for a larger optic size, what size to go in for? Suppose you have higher astigmatism, what size to go in for? And all those things I'll be bringing up in my talk. I'll be also bringing up with complicated situations, the lens opening sideways, the lens rotating, the lens to be explanted, the lens having a vault of 20 microns, all that stuff we will discuss. So I think after saying this, I will go on to my presentation. So am I on?
Yes, Kamal.
Okay,
Yes.
so
Yes, you're on.
okay. So I will be looking at, managing extreme and unique situations managed by using an IPCL. So I guess in last few years, more than 2,200 cases done of phakic lenses. They have taught me a lot. I made a lot of mistakes, learned along the way. So from the age of ignorance, age of innocence, age of arrogance, now I guess all three of us are in the age of Zen. So I'll be talking about a few things. So again, before I start, financial disclosures, I have no financial association with any of the companies of any product I'll be talking during the whole conversation. And I will not, I have no, I am a QL for the companies, but that's about it. I have no financial disclosures. So we have various options in phakic lenses. My main experience I would be talking about on STAR and mainly the IPCL. Though I have used all the other products available in the market, I've used a lot of Apasami lenses, biotech lenses. But if you consider the total market of phakic lenses, I guess these two guys rule the roost. These two people are actually kings in phakic lens technology. So just a brief thing, few basic things in your lens or the star lens or the ICL, the leading hole is always on the right. And on the IPCL, the leading notch is always on the left. So if you audience can actually see this, this is what it would be. Now there are got two holes extra, which Dr. Himanshu talked about. You always need to remember when you're loading this lens, these two holes should actually be facing the superior eyebrow. So this going left and these two holes coming on top, or you're all set to go. And of course, what goes without saying is that the lens always lands with the concave side down and convex side up. I will not get into this. There are different holes. The company claims that, you know, a size of a larger size with a tapering edge gives lesser glare. This size or the spring mechanism of the lens is more resistant to higher volt. In my series, I have patients which are 1180 volts and they are doing perfect for last over 2 years. So that is from my track record. We will see some more cities as we go. So broad statistical analysis, just talking about IPCL, nearly 2016 cases done. V1 model, which was the earlier model, I have a large amount of experience with that. 564 done with V2. Follow-up period of six years. I've done 17 cases of piggyback for post-pseudophakic residual error. Now, this is something interesting. We are going to be opening because I was getting a lot of questions whether can the IPCL be used as a piggyback? Well, yes. This is one of my favorite techniques. I use this very often. Patients with residual refractive errors, patients who want progressive or presbyopic correction after an implant of a monofocal, it's an ideal situation. The best part is doing one surgery where you put in one piggyback presbyopic lens. It can also correct the refractive error which was previously seen in the patient, while it corrects the cylinder and it also corrects the presbyopia. So that's a huge advantage. With one procedure, you're able to take care of your IOL surprise and also the near distance correction. The V1 model, we used to do laser PI and a lot of time we would do iridectomy. I still do iridectomy in my few patients. I will share how I do it and where I do it. I still do not take, even in spite of the central hole, if I see if my angles are not very good, if my iris configuration is not to my liking, or if the anterior chamber depth is less than 3 or 2.9, I still go and do a PI either during the surgery or preoperatively, and I've never have a reason to regret it. And as Dr. Himanshu said, we've corrected up to +6 diopters of hypermetropia. So I had a question from one of our friends that can it be done for hypermetropia? Yes, sure, why not? But the only problem in hypermetropes is we do not get the adequate anterior chamber depth. So if you have a good anterior chamber depth, which by far is not very usual in hypermetropes, because hypermetropes have a smaller axial length and a smaller ACD. But in case you have an adequate ACD, you can go ahead beautifully by using a IPCL or a hypermetropic IPCL. So what are the issues which we have experienced till now? Majorly no issues, no glares and halos. We've done approximately 6 explants. We'll go into the details, and lens exchange in 9 people over nearly 2,000 people, which I think it's a beautiful series. One patient was explanted because post-trauma, he was walking on the road, he got punched in the eye, and he got a cataract. 4 other explants were due to shallow vault. They were shallow vaulted, and the patient got cataract, anterior subcapsular cataract. We had to explant and do a multifocal lens in that. There was a patient with post-RD surgery who developed a cataract, post-RD surgery and silicone oil, but no explants due to the halos. We've done 11 exchanges total, so which means 11, we've done 5 bad oversizing and 6 due to a repeated malrotation. Now, as Dr. Himanshu said, when would you want to explant a lens if it is rotating? In my textbook, if the lens has rotated over 30 degrees, no need to redial it again. It will rotate again. That's my experience. I've had burnt my hands, patient coming to OT, getting operated, going back, the lens rotates back again, and I rotate it back again, and the patient goes back and comes back again after 1 week. So my bottom line is 1 clock hour or 30 degrees. If any lens rotates more than that, explant it and go to the next possible size. And if you have an access to a UBM, you can actually see what is the proper sizing you would want, but it doesn't matter. You can, if the rotation is happening with a low vault, which means you have a vault less than 300 microns and the lens is rotating, you can gladly go in for a 0.5 millimeter larger size, which means each 0.25 micron will add 100 to 125 microns to it. So you will have a vault of 500 to 550, which is just ideal. So if you have a lower vault, go in for not just a 0.25 in my experience. If the vault is less than 400, I would go in beautifully with 0.5 millimeter larger IPCL. Now, redialing, as I said, if it's a small redialing, you go ahead. Now, the best part about this particular lens is, as Dr. Himanshu said, that it is customized and personalized. You do not need to dial this lens to the astigmatism. The lens comes preloaded with astigmatism in it. So what means is all you need to do is mark your 0 and 180. And once you do that, the lens is only aligned at 0, 180 and rest all is taken care of. So the mainstay is white to white ACD, but I must bring in the concept of pupil size. Pupil size measurement is very important. I will skip this slide. Dr. Himanshu's covered it beautifully, but I just highlighted 14.5 because in my series, I've done till 14.5 millimeter of phakic lenses. I believe I've done all kinds of cases possible with phakic lenses, and we will see as we go along. We will just see a brief video. Now, this is a video of how this phakic lens, this is how it comes loaded. You can see the holes. Now the lens goes with concave side
Kamal, the video
down and convex side up.
Kamal, the video is not playing.
Okay, I'll go to stop screen. I think the same issue.
Yeah.
I hope now it plays. Is it playing now? Is it playing now?
It's stuck. Did you click on that OK?
Yes, I did.
Can you click one more time, please?
Let me just switch it off and now go back again. I have done the optimized screen resolution. And can you see it on the screen now? I will just go on.
Yeah, I can see on the screen. Full screen.
Now is it playing?
No.
No, Kamal, it's that only Microsoft Office OK message coming.
There is some problem. I think we'll have to go to the support team and I guess till the time they get the video playing.
Gaurav, can we help?
Gaurav, the video is not playing. Gaurav? Gaurav? Because my presentation is majorly based on the videos. Is Gaurav there?
So, Kamal, meanwhile, you can sort it out, and I and Himanshu would be taking up.
Is it? Can you see the video now?
Yeah.
No, No,
okay?
it's the screen is still frozen. It's like.
So it says opening.
Can you see anything?
No, we can hear the sound from your system, but not on our system.
So I guess now can you see this slide?
Yeah.
Yes, slide is visible.
Okay, so till the time Gaurav tries to help us out with that. So I had a question asking me what are the contraindications for the phakic IOLs? First contraindication would be, first of all, ascertain what is the reason of myopia. It should be only an axial myopia. A lot of times you would get a sclerotic cataract, early onset. We've had patients who approach to us with myopia at 34, onset of myopia. So take the history. These patients a lot of times could be emmetropic to start with. And suddenly they say for last 1 or 2 years, I'm getting myopic. And the myopia starts increasing. Some of them would have maybe -3, -4, and suddenly land up with -8. So always rule out the axial myopia. So always dilate these patients and do a red glow evaluation, again, the retroillumination. Corneal dystrophy are definitely a contraindication for these patients. Anterior chamber depth less than 2.8 millimeters for all beginners. Of course, some of us who've done a lot of cases can go around with cat's whiskers the side. History of uveitis, even if it is healed. I had a question asking me, is it a good idea to put this lens? At least in my hands, I would still say I would avoid using this lens in patients who are have history of anterior uveitis. Now, if the lens rise is more than 600 microns, a beautiful concept explained by Dr. Himanshu, I'll explain it as we move along. If you have a lens rise more than 600 microns and your AC depth is less than 3.2, do not go in for this lens. Presence of posterior synechiae is a partial condition. It's a partial contraindication. We'll see some videos how we manage that. Of course, a patient of known glaucoma, angle pathologies, iris cyst. I've had very interesting cases where one eye, the patient does very well, and the second eye, the pressure goes high, the lens keeps vaulting, though the white to white and everything is fine. We did a UBM to find that the patient had a retro iris cyst. Now, this cyst was pushing the IPCL forward and causing intraocular pressure rise. So you could get these freaked out cases. We'll see videos as we move along. Any history of retina detachment, and of course, a long-standing diabetes mellitus, and of course, very thinned out retina in high myopes is a relative contraindication for these cases. Now, what are the learnings which we have got as we moved along? I've done patients till -34 diopters with 2 diopter cylinder. Patient had a delayed RD, of course, not surgery induced. This patient was predisposed for a retina detachment. After 1.5 years, had a retina detachment. High to moderate, I've corrected till 11 diopters of astigmatism, especially in cases of corneal scars, where with the corneal scar, if correction, the patient improves a reasonable vision. No need to take the patient up for a corneal transplant. Accuracy of the toric lens is exceptional because it's a smart toric lens, as I explained. Once you put the lens at 0-180, it fits in fantastically. Exceptionally large corneas. We realized that this particular lens works fantastic for exceptionally large corneas. But one caveat here, if you have a very large cornea, you must note, does your pupil of the patient also dilate well or not? Now, in case you have a large cornea, but the pupil does not dilate beyond 8 to 8.5 millimeter, and you have a size requirement of 14 millimeters, well, for a beginner, it could be tricky. So pupil size is important thing which we all should be looking at apart from our anterior chamber depth, a white to white, sulcus to sulcus, and the lens rise. This is a new cat which I'm getting into the system. Start looking at the pupil size because your patients who complain about glares, halos, nighttime problems, they will go down significantly if you start mapping your pupil size. I have done pupil IPCL sizes up to 8.5 millimeter. Another thing to note is what is the resting pupil size and what is the maximum dilated pupil size. We will discuss as we move along there. Whenever astigmatism is more than 2 diopters, always try and measure the vertical diameter of white to white also and give it to the company because it helps them decide the size and the vault. So this is something new which we've developed over time. If I have anything more than 2 diopters, I give the vertical white to white also. Now, when you have presbyopic corrections, you can also use a presbyopic lens with these patients and also correct their astigmatism. Secondary piggybacking procedure, I've done a lot of that. We will see some videos. And in case there is a damage or a mark on the lens, you could have another backup lens and explant that on table and remove that. Now, fine touching of the points, again, always dilate these patients. Meticulous refractive error noting is most important. A lot of us are taught that myopes do not accommodate. Well, I've had surprises down the track. A patient reporting with -8 after dilatation was accepting something like -5.5 because just an accommodator over time. So these are the patients. You should always dilate all your patients and do a PMT in that. As I said, maximum possible dilatation. If it's a mid-dilated pupil, watch out. Large cornea, you have to confirm the maximum dilatation. In a keratoconus patient, this is very interesting. If you have a keratoconus patient, the anterior chamber depth a lot of times could be very confusing. Because of the central coning, you might get a higher anterior chamber depth, but in reality, in the periphery, the angle is not so good. So when you have a keratoconus patient, make sure that you mention in your file when you're sending it to the person who's manufacturing the lens that this is a keratoconus patient. Also, always measure the angle size. Make sure these patients are at least 8 months to 1 year post-corneal cross-linking, oblique intacts, whatever you're choosing. Let the refraction stabilize. Now, posterior segment evaluation is very important for these patients because they are the high-risk group. They are more prone to having retina detachments like that any other group. Gonioscopy and angle measurement also is very important. And as mentioned by my friend, Dr. Himanshu Specular. Now, as I said, retina screening is very important. Large size pupil. Now, pilocarpine. If you put the lens in and you use a miotic agent, I would not recommend that because it stops the viscoelastic, if you use viscoelastic, to flow out from behind the lens, and it may cause retained viscoelastic and give you a wrong vault. Putting, as long as the pupil is constricted, it will reduce your vault by 150 to 180. I've documented this. If you put a pilocarpine and the patient is still myosed the next day morning and he reports with 700 micron vault, be sure that you'll get 800 plus micron when the pupil comes to a larger size. Similarly, if you see a patient with a dilated pupil, this vault will go up by 100 to 150 than the resting pupil. So suppose you see a patient in a dilated state at 850 and you start getting worried, oh, I have a high vault, relax. When the pupil will come down to normal size, this vault will settle around 680 to 700. Now, these are learnings which I picked over a few times. Always, if possible, watch your patient first few hours after the surgery in case you've used a viscoelastic, because these patients classically will have an IOP spike within first 1 or 2 hours. In case you have an IOP spike, take the patient, put a fresh drop of Paracane into the eye of the patient, use a 23 gauge needle, just push the side port slightly from the limbus, let a drop of aqueous flow out and the IOP immediately comes down. We'll see a video. Breakpoint is these patients are high steroid responders. Always watch out in the early postoperative period. Whenever a patient is reporting to you with decreased vision, who was initially doing very well, he had a good vision and suddenly the vision goes down, please don't forget to check this patient's IOP. This patient could be a steroid responder with a high IOP. Now, what are the learnings? The learnings, as I said, are preoperative, case selection, and exclusion criteria. What are the investigations to do? The measurements and special situations. Peroperatively, if you're using a toric lens, please make sure your marking is accurate, because if your marking is not accurate, everything will go wrong from the first point itself. Peroperative, there are some special situations which needs to be managed. If the lens is opening down, if the lens is getting stuck in the wound, so we'll see some videos for that. Postoperatively, what are things to be done immediately post-op, intermediate and delayed post-op? We'll be talking about that. So we will just see few case examples very fast. Now, this is a patient. Look at the thick glasses he's wearing. -27 with -3 diopters of cylinder. Post-surgery, this is what we classically are taught, the beer bottle glasses. Look at the thickness of his glasses. And look at the smile on the face of the patient. Of course, I had to cover the face of the patient to take care of the privacy. Now, this is again a patient with -15, with -3, -17 in the other eye. Now, this patient had an exceptionally large white to white. So I used a 14 millimeter diameter lens in this particular case. This is a case of two twins. Both of them got operated on the same day. The sister was -12 diopters. The brother was -10 diopters. Both decided to get operated on the first day. Now, I had a question said, can we operate both eyes on the same day? Well, in my practice, I do not do that. And my reasoning and logic is, if I do the surgery on one day, because as Dr. Himanshu beautifully elucidated, the sulcus and the vaulting is a blind step. By experience, we've finalized the nomograms, but the predictability can be 1-2% up and down. So suppose you land up with a bad vault or a low or a high vault or a rotation, you should have enough time to see. So in my practice, I give a gap of 4 to 5 days between one eye and second eye as a standard practice. That gives me a lot of time to plan my surgery, learn from my earlier surgery.
Kamal, sorry to interrupt you one thing. The support person says you finish the slide and you can play the video separately.
Yeah, I can do that. I can do that.
Please go ahead.
Yeah. So now these are two brothers. They were not as courageous as the earlier brother and sister. Now here, the one brother gets operated. He's very happy. The second brother wanted the elder brother, who's 3 minutes elder to him, is a twin. He wanted to see how the surgery fares, and then he came for surgery. Now, this is a lady with -29 diopters. She had a retina detachment in one of the eyes, but she did very well for 2.5 years. Now, this is a patient with -34 diopters with 2.5 diopter cylinder. This gentleman I've operated around 8 months ago. He's super thrilled. Now, one word of caution for the audience. When you're dealing with such high myopes, you need to be very careful about their pupil sizes. Number one, these patients may not have a good fusion. Their orthoptic test has to be mandatory, because sometimes these patients may report to you with diplopia or glares and halos, which could be no fault of the lens. It could be just that they've had a bad fusion. They're not orthophoric. So make sure you do an orthoptic test, especially when you're doing patients beyond -14 diopters. Though this is not a cutoff, even at a lower myopia, you can have that. But especially like that. So I have burnt my hands doing such cases. Now let us go to preoperative curve. So as I said, refractive correction is the most important. You are needing to note the refractive error of the patient is very important. Role of pupil size measurement, again, you also need to put some NSAID, because for a beginner, if you manipulate the phakic lens on the iris, a lot of times the iris will shut down on you. Use an NSAID before the surgery so that the iris does not close down on you. Different methods of measuring pupil. We can use, if you have access to a machine which is measuring a dynamic pupillometry like we do, we get a scotopic and a mesopic pupil. Like somebody asked me, what are the basic tools, most basic tool a doctor can use to shift to a phakic lens procedure? Well, here's your answer. You can use a simple autorefractive keratometer, which has got marker where you can measure the pupil size or limbus size. So all you need to do is switch off the lights of the room and use the pupil marker on your autorefractometer. Take 3 readings. By default, you will get a correct reading. Now, for the white-to-white measure, as Dr. Himanshu measured, we take the mid-limbus. I will not spend much time on it. My personal take is slightly different than Dr. Himanshu and Dr. Sonu. I use the caliper to finalize my readings. So I use an autorefractive keratometer. I use a Cirrus. I've used an IOL Master. I've realized that the IOL Master gives one of the most deviating readings, a lot of times much larger than I expected. So by default, my Cirrus, that also on an edit mode, which means, as Dr. Himanshu had shown, when you take the image, please recheck it. That has the machine mapped the white to white properly? If not, edit it manually, use your estimation, put the marker line on mid-limbus, take that reading, and use the vernier calibrate only to corroborate your early readings. So in my practice, my vernier caliper is one very important tool. A lot of times, if I get a discrepancy in other machines, I resort to vernier caliper to reconfirm my calculation. You could use a vernier caliper on a microscope. I've used an ARK to measure this. If you can see in this situation, there's a 6.6 millimeter pupil measurement on an ARK and approximately an 11.4 measurement of the white to white. So you can use these simple tools also, but the only thing is you will not get the second decimal. You will not get an 11.45 or 11.48. So make sure you take 3 readings and take an average of this, and it works fantastic. Now, anterior chamber depth, I had a question. What if the person does not have access to an expensive equipment like a Cirrus or a Pentacam or an IOL Master? You can use an A scan, but only on an immersion mode. For that, take 2-3 readings. Most of these machines are fairly accurate. We've tried to do a comparative study of all these. They work excellent. I subtract the corneal pachymetry taken on an ASOCT from the A scan, and most of the times I get a fantastic accuracy as measured by other equipments. Now, as I said, we need to use a manual mode because a lot of times a pigmented conjunctiva or a pterygium could give a white to white, which is erroneous. Now, as I said, in case there is a variance of 0.2 millimeter of white to white between 2 eyes, please go back and recheck again. Change the modality, change the machine, because anything more than 0.2 millimeter variance, especially if the myopia is similar. You can have a difference if your one eye is -12 and the other eye is -2. Yes, this difference can be explained. But if you have similar myopia or a similar refractive error and no astigmatism or similar astigmatism in both the eyes, anything more than 0.2 millimeter of variance is something you need to go back and check again. You can use a UBM in these particular situations. Now, IPCL, ideally, I choose a 1.1.25 larger than the white to white category. If I'm using a larger optic size, I go for a little lower volt. The reason is, suppose for a 6.2 millimeter size optic, I was getting a volt of 700. If I use a larger optic, I would go up by nearly 150 to 200 microns higher. So if you're ordering a little larger optic, go in for a 0.25 lens size smaller, which you would have done for a 6.2. I'll just repeat. In case you're ordering a larger optic size, go for 0.25 smaller than you would have ordered for a standard optic size. This will give you a good volt. Otherwise, a lot of times you could have the pupil just rising up like a plateau because of a large optic size of the. Now, somebody asked me how to fill the form and what all to note. This is all that we note. Now, one thing we do extra is we take axial length and biometry of all these patients for our own calculations and sending to the company to fine tune the results. It gives us 2 or 3 extra information. It confirms our axial myopia. And also, God forbid, if this patient ever has a cataract, I've done the biometry already. It is in my records. I can go back to this biometry, though we will touch that topic as we move along, and this can be used. Now, perioperative learnings. Always achieve maximum dilatation before you take the patient inside. When you're loading the lens, make sure that the orientation is right. The left leading or the right leading, whatever is the case, if it's an ICL or an IPCL, is right. The loading has to be proper. Always open the lens on iris. A beginner will be tempted as the lens is opening in to push it under the iris and just end up, putting the trailing behind the iris. Well, no, this is asking for trouble. Always open the iris on lens on top of the iris. Avoid manipulations. Another important point is do not overinflate with viscoelastic, especially in high myopes. If you overinflate with viscoelastic, the iris will go back and touch the crystalline lens. Well, now you're in trouble. To push the lens in on a posterior back vaulted iris is very tricky. So do not overinflate the viscoelastic. I will show you videos how to manage that. Avoid touching the center part of the phakic lens. This is the thinnest part of the phakic lens. If you touch it, a lot of times it could be something like 70 to 80 microns. You touch it, you might cause cataract and then end up blaming the lens for it, whereas the surgeon is to be blamed. Ensure proper unfolding of the lens in the iris before you finish the surgery. Make sure you rotate the lens 20-30 degrees each side to make sure that the lens haptics have unfolded behind the iris. And of course, meticulous viscoelastic removal is very important if you use viscoelastic, because if you do not remove the viscoelastic, you'll get an erroneous fault. You may get a TAS or a reactive reaction in the anterior chamber, of course, on an intraocular pressure rise. And try possible do. So this is a marking which you can do. I will not repeat it. This is how I do marking on slit lamp. You can use this primary and secondary mark, but the best part is with the IPCL, you do not need to do a secondary mark. All you need to do is you just place the lens at 0, 180. As you see, the marking has been done. The lens has been placed. These marks are at 0 and 180, and that's all you need to do. Now we'll see a small video of slit lamp marking. Make a slit beam to the widest, 13 millimeters. Orient it vertically. Make sure that the patient has it straight and looking into the light. Align the light straight into the axis. Align the axis. And then there you align the axis, and there it goes. Once you have these two marks, all you need to do is go in and place the lens according to that. We will not touch about vaulting. An ideal vault is anything between 300 to 600. The distance between the anterior crystalline lens and the posterior surface of the lens. Let me repeat again. The posterior surface of the phakic lens and the anterior surface of the crystalline lens is the vault. You could have different vaults. Now, if the lens is too short, it will cause rotation. If it's an astigmatic lens or a toric lens, you have to explant it. If it's a non-toric lens, Dr. Sonu explained it beautifully. Just rotate it vertically. And if it holds true, fantastic. If the lens is too long, it will cause crowding of the angles and may cause glaucoma. Now, this is called lens rise. The lens, you draw a straight line from angle to angle, measure the distance of the crystalline lens rise from this line. If it is more than 600 microns and the anterior chamber depth is lesser than 3.2, this could be a tricky situation. Either avoid this case or go in for a higher than planned vault. But in case this iris lens rise is less than 600, you're okay to go. We don't see such kind of cases very frequently, but yes, you would come across these cases. A good way of measuring the vault on a slit lamp would be this condition. One slit thickness of a cornea gives a good idea. Now, post-operative, visual acuity to be measured on all visits. Measure the intraocular pressure. This will give us an indication of the angle closure. On slit lamp, see the angles and the iris. It could be a retained viscoelastic, and if it's delayed, it could be a steroid responder. Vault, in my practice, we see on the first visit and the last visit. And then whenever the patient visits us again, I have a classical case of a girl whom I operated 7 years ago. She shifted to Australia and came back to me after 3 years. Her last vault recorded with me was 470, but after 3 to 4 years, she came to me with a vault of 20 microns. And on history taking, I realized that she developed some eye allergy there, and she was frequently rubbing her eyes very hard. So one eye vault had gone to 134, the other eye vault had gone to 20 microns. So we went in and explanted those lenses, and we told her that if you're going to be staying there, you need to be on anti-allergics and low-dose steroids. As I said, if you used a pilocarpine, you could get a lower vault or cause a high IOP. If you see an early retrolental opacity, do not worry. Suppose on the 4th, 5th day, you see an opacity behind the lens. Do not panic. This is not cataract. You will not get a cataract so soon. Best thing is to do an anterior segment OCT, and just behind the lens, you'll see an opacity which is not in the capsule or subcapsular. It is anterior to the capsule. This is retained viscoelastic, which should disappear over time. Now, in case the patient reports with drop in vision, look out for a rotation if it's an astigmatic or a toric lens. Look out for an intraocular pressure and look out for retinal pathology. It could be a macular pathology or a retina problem. So post-operative follow-up, these you need to do. Now we will see some interesting cases. This is fourth day. In first case, it looks like anterior subcapsular cataract. And then after a few days, this starts clearing up, and the ASOCT also shows the same. This is a lens with varying vault. Now, this is a very interesting case of a toric IPCL in a keratoconus. Now, as I mentioned, this was our learning lesson. We got a very good anterior chamber depth. Based on that, we decided the phakic lens without measuring the angles in great concern and see what we landed up with. The AC depth was erroneous because it was only deep in the anterior chamber due to keratoconus, and the peripheral angles were not that good, and we landed up with an angle closure glaucoma, and this lens had to be explanted. Now, this is a cataract with there's a low vault. This is an anterior subcapsular cataract. It has appeared after 2 months of surgery, and I'm checking whenever you see an opacity, always check the anterior segment opacity of this patient. You will find a very low vault. Now, this is a clear-cut indication to explant it. Now, you can wait if the patient is not much bothered by it, but I would say if you started seeing a cataract in the early stage and the lens is low vaulted, so the lens keeps rotating, may cause some pigment or iris chafing. So it's a good idea to go in and take it out earlier. So we had one question to when is the good time to explant a lens? In case it has started giving visual disturbance to the patient and the lens is rotating freely, I think it's a good indication to explant the lens. Now, these are peroperative learnings. This is an 8.5 diopter. I hope this video plays. Can you see the video?
No, the video is not
there.
Video is not playing.
You have to play it from the VLC player, and then you have to set.
No, it's playing in the VLC player, Rushabh. My concern is it is playing in the VLC player. I will try and do the same procedure again.
Well, you can play all the videos separately. That would also be fine. One by one, if you can take up. And
I can do that, but Himanshu, my folder has got gazillions of videos from where I'm taking it.
I understand.
So I'll be hunting for them. Let me do one more trial. It always seems to work in other places. Still, do you get the video?
No.
No video.
No.
Hey, what does it say?
That OK thing which you have to press, it's just stuck there.
Rushabh, can you help me? Rushabh?
Yes, sir. You have to play the video from the external devices.
If I'm going to play from the external device, okay, then what I'll do is I'll just try and hunt for them. Meanwhile, I'll just finish my presentation.
Okay, sir. Sure.
I guess let's do that. I will try and hunt for them point by point. So, because now it's just videos, all interesting, crazy videos. Now, this is the lens where there was overinflation with viscoelastic. The chamber was 3.4 millimeters. And this video shows that if I've overfilled with viscoelastic, the iris practically, the pupil disappears. The iris goes actually practically vertical, and I cannot even see the pupil edge. So it's very difficult for me to push the lens in behind the iris. So the trick which I wanted to show the audience is I burp the wound, get the viscoelastic out. Now, I've recorded these videos especially by creating these situations for audience to realize that how much of problem it can cause even an experienced surgeon, that if your AC is too deep and there's a lot of viscoelastic, you can get stuck up. Now, this is a 13.5 millimeter large size pupil, 13.5 millimeter IPCL, but the pupil did not dilate beyond 8 millimeter. What problems it can cause for a beginner surgeon and how to manage that, I will be showing in this video. Now, this is again a video where the lens has been loaded upside down by my assistant, though technically I always load the lens myself. Sometimes I have my assistant load that. But this was one of those bad days. So this is to show the audience how easy it is to explant the lens in the same sitting. Now, I had a question from an audience that can the same lens be reimplanted again? Yes, sure. Why not? If you have not broken the lens or damaged the haptics or the central optic, you can put the lens in again. In this video, we will see that once we go in, As the lens opens, we just hold one edge with the McPherson forceps and it comes out easily removed. Now, this is an iridectomy for a patient where I was having a high lens rise. Again, I will show you the video how this iridectomy is done using a side port and selective pilocarpine in one area and selective viscoelastic in one area. This is an amazing video. This is the technique I do. Can this video be seen?
Yes, it's visible.
Yes, we can
Clear.
Okay, now this is a patient who developed a IOP spike. We have the needle, 26 gauge needle bevel backwards facing me. Ask the patient to look upwards, put a drop of Paracane, and there you see I've made the aqueous leak. There, can you see the drop come out?
Yes, we can.
Yeah, you just depress the scleral lip, have one drop of aqueous come out, and in 30 seconds, the cornea becomes absolutely clear. Now, this is one patient which was operated by one of my associates, and you see the cornea has become clear immediately there. Immediately. This case was operated by one of my colleagues. Probably the viscoelastic was not taken off well. So I was just called, as a habit, I call all my patients immediately post-operatively into the OPD before they sent home. And I noticed that the cornea was getting hazy and the patient was getting headache and nausea. No need to have this patient wait or give him some acetazolamide. Just burp the wound using a 26 gauge bent needle on a side port with a slight pressure and the aqueous will drain out. Now, in case immediately post-op, you see a higher vault, relax, don't worry. This is a common condition a lot of times seen if there's a retained viscoelastic, if there's no high pressure and you see a higher vault. A lot of times your normal gut instinct will tell you that the iris may close behind the IPCL. It will not. It tends to override it. So just let it be. Make sure you keep this patient on a follow-up. Do the ASOCT on the first visit and one after 1 week. If everything is fine, just forget about it. So this is for beginners again. If you see such a situation of a very high vault, probably even having the iris fall behind the IPCL, do not worry, let it be. Do not try and put pilocarpine in these patients or a myotic because then it will retain the viscoelastic behind the phakic lens. Let the pupil be large, the viscoelastic will flow out and it will settle down faster. Now, this is again a video. I'll try and go in the video section. I am sure it will not run, but my mind says I can maybe attempt once and go and do that. So this is a hook technique which I use to explant the lens. This video demonstrates it is so easy to remove a phakic lens, both an ICL and IPCL. Earlier, the V1 model, you would probably need to enlarge the wound to 3 millimeter to explant. But with this V2 model, you can explant it through a 2.8 millimeter without any problem. The same goes for an ICL. It is very easy to remove the ICL in one go. Make sure you've done your specular before you start removing it. You put in viscoelastic on top and also viscoelastic underneath. So when you put viscoelastic underneath the phakic lens, it automatically rises the phakic lens into the anterior chamber. So this gives you enough space to maneuver your instrument behind the phakic lens to get it out. Now, always put slight amount of viscoelastic first on top and then put underneath. Because if you put first underneath and then put on top, this will again push the phakic lens back in and you will have trouble negotiating. So put slight amount of viscoelastic covering the whole endothelium and the anterior chamber. Now go in behind the phakic lens and put viscoelastic. While you're doing that, make sure you dip the scleral lip to let the extra viscoelastic come out, lest you have raised intraocular pressure. So as you're pushing viscoelastic behind the phakic lens, dip the scleral lip, let the extra viscoelastic come out. And as this lens rises into the anterior chamber, go it with a blunt instrument and just tip it into the anterior chamber, hook it and get it out. We will see the video. Now, this was again a case which was rotating again and again and again. Now, once this patient was rotating again and again, this is the girl, she had 140 micron and this was rotating again. So I had to explant it. Now, this is a mal-rotated lens, which while inserting the lens rotated. So how to manage the lens as it is going in and trying to open upside down. That video shows that. This is another video where there is an asymmetric vault. As the audience, Dr. Himanshu, can you see there is an iris capture superiorly?
Yes, we can see that. Yes.
And inferiorly, everything is fine.
Yeah.
No, in the dilated state, I did an ASOCT and I found a difference of vault in two planes of 340 microns. The lens in the inferior area was 700 and on the superior area was 1040. So there was no reason this should have happened. So I estimated that one of the haptics has not opened properly. All I needed to do was take the patient back into the operation theater, make sure I put a lot of viscoelastic on top of the IPCL, push it downwards, and then just move it sideways 5-10 degrees each way, and immediately the lens settled down. So these things I'm discussing because these things are seen in everyday life. Now, this is again a lens which is rotated by 20 degrees and it is inducing this refractive error to this patient. And the patient came to me with glare and 6/9 vision. If, as I said, 1 to 2 clock hours, rethink. Now, this was video. I just redialed the lens, let it be, and kept the patient on follow-up. Now, this is patient again, beautifully centered at 0-180, and the lens never rotated again. Now, this is an interesting case. I had a question: if a patient gets a cataract, what to do about the biometry for this patient? Because in case you need to explant the lens and do IOL in this patient. Now, my first advice to all is when you do your phakic lens, do a biometry and save it in your record. Number one. Number two, if you have an access to an optical biometer does not give any difference between a phakic lens and a normal patient. So we've done a few cases and I've realized it doesn't matter. In case you're using an immersion scan, there are some formulas for that. Normally, in case you don't want to use a formula, just buffer it up by a 0.75 towards myopia and you'll land up right. Now, this is a formula in case people want to note it. If you use the axial length used at a 1500 meters per second ultrasound speed plus X. Now, X is a factor or it's a constant which is 0.10 for a Collamer and 0.23 for an acrylic lens. Use this and multiply it by T. T is the central thickness of the IPCL or an ICL, which an average is anything between 80 to 110 microns. You could get your value of corrected. Now, these were some very, very important situations. This is a video where a patient has got a symphony done, operated elsewhere. He comes to me with a 4.5 diopters of residual hypermetropia post-symphony. The earlier doctor sent the patient to me for lens explantation and putting a new symphony. But I must share with the audience, for last so many years, I've stopped explanting lenses. For any residual refractive error, I just use a piggyback IPCL or a piggyback lens. I have stopped causing trauma of cutting and damaging the iris or endothelium, damaging the endothelial cells. I just use a piggyback procedure. So this was a video where I show using of a plus 4.5 IPCL in a patient with symphony who was 6'6" within 1 hour of surgery and very happy. Now, this is an interesting case of corneal scar where the refractive error was minus 3.5 and the patient had 11 diopters of cylinder. I operated this patient and this patient landed up with uncorrected vision of 6/12 parts and was more than happy. So we did not have to, this was again a video. This case was operated by me for corneal perforation with a beer bottle approximately 10 to 12 years ago. And then he developed cataract and then he had a cataract. Then the cataract had a residual astigmatism. So all those things crept in. Now, this is again a case of central corneal scar with -4 and -5 diopter cylinder. Now, the best part about this IPCL is, if you notice, all you need to do is align these lenses at 0 and 180 and your astigmatism is taken care of. You do not need to rotate these lenses to different axes or do gymnastics. So I feel in my astigmatic patients, my case selection is very, very easy if I decide to put in this lens. Now, this is a patient operated by me 18 years ago. This patient developed a -17 diopters of myopia. Now, this was a young boy. He got hurt when he was young. I operated him for a cataract 17 years ago, and he had a -17 diopters. Now, when he comes to me with the refractive error, I cannot correct -17 with a LASIK. I could have put a phakic lens, but then I realized there are posterior synechiae all over between the bag, and the bag has fibros. So if I explant this lens, I'll be explanting it with the bag, and I'll be needing to go it with a sulcus fixated lens. So I decided to do this surgery. Again, I'll try and show this video. I decided to do this surgery by doing synechial. I cut the synechiae between the anterior capsule and the posterior part of the iris. I create a space and I put this. And this patient was thrilled. He had 6/9 vision. Now, this is a post-operative case. Now, I had a question from one of the audience. Can we put a piggyback lens as IPCL post-YAG capsulotomy? Yes, you can. If the lens pseudophacos or the IOL is in the bag from the previous surgery, and the bag is well captured all sides on the IOL, and there is a small YAG capsulotomy done in the posterior capsule, and the chamber is stable, you can actually go ahead and put a piggyback lens on top of your earlier pseudophakic eye without any fear of the vitreous coming in. Now, this is again a very, very interesting case. Again, this was lens customized. I think the smallest possible lens ever manufactured, a phakic lens. It was a 4.5 millimeter small minuscule optic lens of a 13 millimeter and -13 diopters. This was a non-small dilating pupil. It was a complete fibrosis of the bag. There was a posterior YAG capsulotomy done in this patient. Complete posterior synechiae between the anterior capsule and the iris. High refractive error of 13 diopters and a thin cornea 496. Guess what saved the day? I used a customized personalized lens made at 4.5 millimeters. The reason being, there was so much of synechiae between the capsule. I was not sure I will be able to put a 6 millimeter lens and it would fit well without angle vaulting. So we went in for a small lens. I hope I'm able to show this video as we move along. Now coming to another interesting topic of presbyopic phakic lenses. I've had my share of presbyopic phakic lenses. My first choice of these lenses is as a piggyback, as a secondary procedure. Though I have done it as a primary procedure in patients who are 50 years old and above. But my first choice is doing them in patients who have been operated earlier for the monofocal lens, and now the patient wishes for a multifocality. One of my classical patients, one of my first patients is my OT technician, whom I operated 14 years ago with a monofocal lens. And he would always, a lot of times, have trouble adjusting with his glasses, giving me the right instruments. So at one point he said, sir, you are operating these lenses for everybody. Can you operate me and put a lens in my eye which makes me see near and distance? So I decided to put my first progressive lens or a presbyopic lens piggyback into my OT technician. And it's been 6 years now, and he gives me the right instrument. In fact, so many times due to my presbyopia, have trouble bending my capsule lexus needle. He does it perfectly. He's got a bilateral presbyopic piggyback IPCL in his eyes. Now, these are available in sizes of 1.5 diopters, where it gives a 0.8 diopters of intumate, 2 diopters, 2.5, 3 diopters, and 3.5. Now, the best part is, apart from the near ad, these lenses can be customized to correct any astigmatism in the patient and also any residual refractive error which the patient has from a previous surgery. So it is a single solution refractive correction, especially for my patients who are being operated earlier, and it works fantastic. Now, the sizing is, it will have rings depending on 5 to 9. It's a diffractive design. In 1.5 diopters lens, you have 5 rings. And as the number of the near ad goes up, you have higher number of rings. It's a trifocal lens technology, energy distribution of 50, 30, and 20 for far, near, and intermediate. The diffractive structure is around 3.5 millimeter, and center 1 millimeter zone is there. Now, patient selection criteria. Now, this is interesting. This is a lot of times confusing. A lot of people ask me. I've just done 7 cases with this, but it works well. I always try and do one eye of the patient who's at below 48 years old. Correct the non-dominating eye for that particular near ad correction. I tend to bump it out by 0.25 higher. So suppose a patient requires 1.25, I would correct him by 1.5 for near ad, not for distance. I'm not changing the emmetropy of this patient. I am overcorrecting him by 0.25 for near. So this gives me a long range of 5 to 6 years. This patient can work with monofocality for near work. And after 5 years, when the patient comes back, you can correct the second eye, and then the patient is good to go. If the patient, in my practice, I would recommend, if the patient is more than 55 to 58 years old, I would refrain from doing this as a primary piggyback lens. I would rather take this patient up with a multifocal clear lens extraction or a pre-lex and correct this patient. So my first choice of a progressive presbyopic lens would be in patients who have got a previous monofocal lens and they want a multifocality now. Now, this is an image photograph of my OT technician. We use an Acryos AO lens. I hope you can see Dr. Sonu and Dr. Imanshu, the classical shape of an Acryos AO lens.
Yeah, we can see that.
And I've placed a multifocal lens with a 3.5 millimeter optic zone, bang in the center. Now I have patients, we have more than 1100 microns. This is 292 microns, patient doing fantastic well, 864 microns, 1000 microns. So the number of holes in the IPCL offer a big advantage. There are 6 holes in an IPCL. Even at such a high wall, this is a patient 2-year-old doing perfectly well at 1000 microns wall. And I think after that, I'll say thank you, and I'll try and show some videos. I hope it works.
Thank you, Kamal. And it was really wonderful lecture as usual. I mean, you have such vast knowledge about all these abnormal cases. And when you bring it to us, it certainly baffles your mind that you certainly can do something for everybody. In,
do we have a video now?
No, we still don't have the video.
Rushabh, I'm playing the video straight from the player.
Yes, sir.
You have to stop sharing the slides, I believe. And
then start again.
Yeah. Maybe optimize.
Share your entire screen.
Yeah, now it's
great.
Wonderful.
Okay.
You can start.
Now the audience will see, I'll go back. I put in some viscoelastic. This is how to explant the lens. So I put some viscoelastic on top. Please remember, I have not overinflated the anterior chamber. Just a slight amount of viscoelastic on top. Make sure it just goes in just enough to push the phakic lens down. Now I'm going behind the lens, and can you see the wave come up? The wave lifts the lens in one direction. I go behind the lens and slowly pull it into the anterior chamber. And while I do that, I make sure that I'm not rubbing or touching the endothelium. Once this is done, I use a blunt instrument again to unfold the lens, and I hook the lens, my blunt instrument, into the hole of the lens. You audience must note this. What I do, I hooked it into the hole, and there I pulled it out. I'll repeat it again. Himanshu, could you see that?
Yes, Kamal, we could see that. We could see that nicely.
I've got the lens into the edge. I let it come towards the wound by dipping the wound, and let viscoelastic come out. Along with the viscoelastic, the lens comes out. It's such a thin and a stretchable lens. Now, in case you want to push this same lens back inside, I would recommend use 2 instruments to pull it like a tug of war rope. Okay, so I'll again go to some other video. Now this is again, can we see that? Can we see that?
No, sir. Go on the top, new sir, and select your video and then play.
Before, while he is loading, remember, when we want to put the same lens again, the scenario is when it's opened on the wrong side or something like that.
Is it playing now?
Yeah, now it's open.
Okay. Now this is a piggyback lens in a symphony. I could have explanted the lens and put in a new lens. All I did was I got a lens manufactured. Now people say that the anterior chamber depth could be an issue in a pseudophakic lens. Well, no, because in pseudophakic lens, your ACD is very, very good. You cannot go wrong. Now I've inflated the lens. I've loaded the lens here, therein, and And there I push the lens on top of my multifocal lens. And putting this lens is very easy since you have a very deep anterior chamber. And there you go in. And there the surgery is over. We'll see more videos. Let's see. Does it still show on your screen?
Yes, it shows. It shows.
So I guess this is a good idea to play one video at a time. Now, this is an interesting case which I'm going to show. We had a patient sent to me by my retina surgeon. This is a case which I will, very interesting. My retina surgeon comes to me and says that, sir, you operated a patient 2 years ago. She had a retina detachment. We did a silicone oil RD surgery. Everything is fine, but now she has developed a cataract. So I see the patient on slit lamp. It looks like a cataract. So I'm prepared to remove the lens and do a cataract with an IOL in this patient. But before I proceed, I do an ASOCT. And on ASOCT, I find a very interesting thing. Now, I want the audience to see this. You know, Himanshu, this is what, you know, intuition and common sense is made of. See what happens. I make a side port. You can see a cataract, anterior polar cataract there.
Not playing.
Kamal, the video is not coming. You'll have to exit out and then go in again.
Coming now?
Yeah, it's coming now.
Okay, now this is a patient coming with anterior capsular cataract sent by my retina surgeon. So before I decide anything, I make a side port and I just push BSS behind the lens through this hole. There's a hole on the top. Can you see what is happening? This is retained membrane and silicone oil. And there was a membrane just retrolental. Can you see the membrane slowly clearing off?
Yeah, we can see that.
And by the time I'm through, there, can you see the membrane peeling off?
Yes, we can.
And the patient was saved from explantation and a cataract surgery. We washed the membrane and see the membrane has come into the anterior chamber.
Yes.
And the surgery was saved. Now again, we will.
So while he's loading, for a piggyback lens, Sulcoflex is certainly a great lens. Cost-wise, when low power you are trying to put, the Sulcoflex works very well. But whenever you're dealing with high power surprise and toricity, with high power surprise, phakic IOLs like ICL, IPCL can certainly do very well also. So both options are there. Whatever suits in your hand, whatever financially you feel comfortable, you surely can go ahead.
Now I am just trying to, you know, there are so many videos here. Now this is an interesting case. Again, is it visible? No.
Not yet.
Yeah. Not yet. So again, I'll get it. Now this is a patient, that poor girl who got a cataract. Now this is with the V1 model. Now remember, V1 model needs a larger incision to be explanted. Now I was in a fix. This is a girl with -29 diopters, very, very fragile sclera. So I thought if I extend the wound first and explant the lens, I will have to suture the wound and shift to a new place because the wound will be leaking in a high myopic case. So I proceed to do the whole cataract surgery with the IPCL in the eye. Now this is, I think, probably one of the few cases. I've done 2 cases like this in my retina detachment cases. The whole capsular rhexis will be done under the IPCL. I'll just go fast. I put in viscoelastic underneath the lens. Can you see that?
Yes, we can see that.
There, my rhexis is being done under the lens. I've completed the rhexis. There. I do my hydro procedure. I finish the rhexis with the capsular rhexis forceps. There, I aspirate the whole cataract with an IA probe. Once this is done, now not much is required. I explant the lens out, finish the whole thing, put in the lens, put in a new lens, and the surgery is over. So this is what, things which are possible. I just want to show one or two interesting cases where the Sanique were broken. There's so many videos, I need to look for them. Meanwhile, I think you can discuss something.
Sure. So the part what we were discussing earlier about pilocarpine. Pilocarpine, if you're using, intuitively, a lot of neurosurgeons like to use pilocarpine because they don't feel comfortable. As Kamal was saying, even if you're using visco, it retains behind. But even if you're not using visco, what happens? It leads to rotation of the ciliary body, and that reduces the vault. Right. So when you are dealing with low vault, pilocarpine actually works pretty against your intuition. So unnecessarily do not try to do that. It's a very forgiving surgery. In terms of lens, is very robust. I mean, you can remove the lens and you can re-put the lens again without any problem. However, before you put the lens, always observe the lens under microscope. There are no problems which you have caused by removing the lens. The only time where you put the lens is where you had removed the lens for wrong direction, as in flip lens. But if you remove the lens for damaged lens, or whether it's too loose the lens and it's floating around, then it's not a good idea to put the same lens again. I think Kamal is ready with
Okay, now this is a patient where I was anticipating an intraocular pressure rise. So I'll show the audience one method to do a peripheral aidectomy from the side port. I put in a small amount of pilocarpine. This was a patient who had a little bit of abnormal iris configuration. Now put in a bit of pilocarpine. Now see what I do. I put in viscoelastic in one area. The pupil is small. I've done the viscoelastic removal. There, I'll fast forwarding. Now I've stopped. I put viscoelastic only in that area where I want to do the PI, which is just under my side port. I take a cutter, 200 millimeter of vacuum, 100 cuts, and tilt it sideways, slide sideways, and give a short burst. And you will see you have a beautiful PI. So it works excellent. You can also use a central part. Suppose you want to use the main wound. This is what, is it video? Can you see that?
No, not yet. Not yet.
Not yet. I think I've mastered the technique now. Does it show now?
Yeah, it shows. Yeah.
Now this is, again, I want to do a PI. Now how to use the main wound? Make sure you put in viscoelastic. You have to make sure that the pupil really goes small. Again, viscoelastic on more so in that area where you want to do a PI, so that you push the iris backwards. You don't hurt the endothelium. Go from the side port, small little cut, and you realize once it's over, you have a beautiful 500 microns of a PI.
So one tip for those who want to do an intra-op PI, whether it's for combined surgery like DSEC and cataract or anywhere, remember to pay a lot of attention to your vacuum. Kamal rightly said that he would keep 150 to 200 millimeter vacuum. If you falter there, I have seen people where they've sucked up half of the iris and then you are dealing with a huge iridodialysis. Keep your cut rate minimum, and there are going to be little few drops of blood which will come out. The trick to remove the blood is do not keep on just running after blood. You can put a little bit of air inside, and air tamponade stops the bleeding for permanently. So do not run after clearing the blood. It will stop at one point of time.
I think I've got one video which is very interesting. I hope this plays now. Now there. Are you able to see this?
This is
Yeah.
video.
Okay, now this is, as I said, minus 13 diopters, operated 12 years ago.
Presence of severe
You can see it's a non-dilating pupil, complete fibrosis.
Yes.
Total posterior Sanique, 13 diopters, 419. So I go in. First I break the Sanique, try and dilate.
doctor's manipulator.
I stretch the pupil,
of the iris.
break the synechiae.
Under the
Now I'm breaking the synechiae.
zone. Now this is a
Now
very
this has to be done carefully. You will cause a lot of hemorrhage.
Do this and do not break the synechiae, cylinder.
Now the best part is not the surgeon, it is but now I do a little bit of pupilloplasty. I call it microsphincterotomy pupilloplasty. I give small nicks all over because this patient's resting pupil size was less than 1.5 millimeter. So I've given the patient slight. Now this is, you can see, a 4.5 millimeter
13 diopters of myopia
phakic lens, which is so small.
would have
There.
probably made
It is a 12 millimeter long and a 4. Can you see, Dr. Himanshu, it's such a thin lens.
caused problems.
Yeah,
Small lens.
we can see that.
We do
Can
that.
get caught up or
So what I'm trying to say is,
silicone plunger
phakic
punch
lens
as
is a
to make
possibility,
sure that
offers
doesn't
so many
happen.
possibilities to us as surgeon.
Viscoelastic force
In
and
areas where you thought you
that
would
enter
hit a
the
stumbling
tunnel
block,
of the
you can
cartilage
actually do these
before
surgeries.
the
You
flanger
can actually
or
help
the
these
plunger of
patients.
the
The only
silicone
thing is you have to be very careful
goes
when you're
and
loading
right
such a
over the
small lens
lens.
because your
Once the
silicone
cartridge
bunch
is
will
loaded
tend to
onto
override the
the
lens and will break the
injector,
lens. So
we
I have
take,
injected the lens
unlike
through
a
with the
normal
viscoelastic and
situation
see
of approach
how
and
beautifully
make
the
sure
lens goes in.
this
This is a
because we
4.5
never
millimeter
knew
lens.
that if the lens, which was
I use an instrument
high
on the
voltage
top.
and longer than the desired length,
It's a long
if
lens.
it had opened in the anterior chamber, because if that were so, it can aspiration with the coaxial cannula, can aspiration left mapping away.
There, the lens is in. And the patient is doing wonderfully well.
Thank you.
This is post-op. So there are so many videos, I guess I'll skip.
It's very nice, Kamal. I mean, it was such wonderful to see plethora of cases which you have, and we wish that time never stops and we just keep on going. Unfortunately, at one point of time, we have to stop so that we can have next one more time and one more lecture, which we can come out with the same topic, but in a different way, and we can take more inputs and know more questions. So
Okay,
again,
so we
can go through the questions.
I must
I
congratulate.
think we all will review the questions once before we close in.
Yes.
Now, IPCL newer version coming or not, that we are not the right people to answer. The company will. Uveitis contraindication, yes, we've discussed, at least with me.
Yes,
Dr. Himanshu, what's your take on it?
So I absolutely agree with Sonu that any such kind of condition, uveitis, when you are keeping a lens on ciliary body, remember uveitis is not just uvea as an iris. It can affect the ciliary body. It can have membranes there. It can have inflammation there. It is not smart thing to put a lens which irritates ciliary body, and certainly not, I would suggest. And Sonu rightly said in the beginning that no for such cases.
Okay, so as we said, simple instrumentations to do white to white. I think both of you beautifully explained it. The biocompatibility of materials, Dr. Himanshu, I'm sure you left no confusion there. Both are as biocompatible. Piggyback lens after yak capsulotomy, I think I explained, if your capsule is all over the optic, covering the optic, the lens is well captured in the bag, you can go ahead and put a piggyback lens there, no problem. Is yak capsulotomy a must? I think not required with the lenses today. In few situations, I might want to do a yak capsulotomy. What is your opinion, Dr. Himanshu?
So I would decide based on how is my PC. If I have significant PC on that's leading to some refractive error, my reliability of refraction would be a little poor. And in that case, before I put a Sulcoflex or any add-on lens, I want to be very sure what my refraction would be. In such cases, I would do a small yak capsulotomy, as Kamal said, that's not a large one, the small one, which will allow me to have a more refined refraction, and then I'd go it. But most of the time, if it's a clear capsule, no, I would be fine.
Okay. Biometry post-pseudophakic, I think I've discussed that. Before removing, it's a good idea to do a biometry in the first go. Yes. If not, an optical biometer is the second choice. Worst to worst, you use an immersion biometer. You could have a formula for that, or just use a buffer of 0.75 millimeters diopters. Your take, Himanshu?
Absolutely. I totally agree with you. In fact, all the refractive surgery, you must do biometry. I mean, we love to take all the responsibility on us. Even if somebody has progression of myopia, if you're not documented biometry or axial length, how will you claim there is a progression? You ultimately
Absolutely.
end up
Wonderful.
claiming that it's a regression, it's my fault, machine's fault. Well, that's not required. Every refractive surgery workup must include biometry and axial length measurement. That's absolutely must.
Dr. Sonu, your take on it?
Yeah, I fully agree with what Himanshu said. It is a must. It's a must.
Okay.
I
Now
think we have
coming to the
taken.
sizing, I think we in such detail. I think most of the audience can write. So we had maximum number of questions. Dr. William, Dr. Suez, Dr. There were so many people who were asking about sizing. So we took a lot of time doing this sizing part. Explantation indications, of course, were touched by Dr. Himanshu. The lens is rotating. It is causing cataract. It is causing iris chafing, or the lens is too overvolted, causing angle closure. Those situations. Late and early complications were asked. This was Dr. Artyom Matveyev was asked this question. I think I tried to cover that. Standard deviation between sizes, Dr. Himanshu, I think you covered it, and Dr. Sonu also covered it.
Yeah, so
There's not more than 5 micron size difference.
EMC is like just 5 micron.
Now, sulcus to sulcus question again was answered beautifully by both of them. Sulcus to sulcus measurement.
Just one part, that whenever you're using one particular lens, when you want to use another lens, not like Kamal was showing those 30 adapter, 35 adapter. If you are comfortable with the ICL, and if you suddenly try to use IPCL for those extreme cases, remember the reliability of your sulcus to sulcus and white to white correlation is very poor in extreme cases. So Absolutely. You may end up in complication, and then you end up blaming the lens that, you know what, it doesn't work out, the size does not come proper. Size of lens does not alter in whatever situation. It's your measurement which was a problem.
Okay. Now there's another interesting question: how to fix a dilated pupil, by Dr. Mohammad Abdul Rahman. How to fix a dilated pupil after phakic IOL. So I think Dr. Ramar Agarwal's technique of single pass 4-throw technique is a single throw 4-pass technique. It's wonderful. You can use Seepser's knot. In my opinion, you could do that with the phakic lens there to protect the crystalline lens. You can go in and make 2 sutures on the side and reduce the pupil size. Your opinion, Dr. Himanshu?
Yeah, so there are a couple of things I would look at. If it's not compulsory, you don't go by what you feel. Go by why patient needs it. Now, if patient has glare issue, patient needs such kind of thing, yes. If not, remember that these high myopes are potential cases where they need to be seen for their peripheral fundus. They may end up in some retina issues. You just don't want to go ahead and make pupil really small. So if it is certainly required, as Dr. Kamal said, we can go ahead and we can put sutures to make it small. But if not, just decide what patient needs, not just based on what you feel would be required, more for a cosmetic purpose.
So
I think wonderfully answered.
I would like to point out, maybe he is asking for a situation where after the implantation of the phakic IOL, you end up with raised pressures, and then you have typical that sphincter atrophy.
Yeah, Urate-Zavalia syndrome. Sure. And
so those kind of conditions, right? So I had these 2 conditions with me, and once was because of the PI not patent in the V4 model, V4B kind of situation. And after making the PI patent and controlling the pressure, the patient had a fixed dilated pupil. And I think, as rightly Kamal said, the 4-throw pupilloplasty in these cases works very well. And then you can use that modified ICAMH technique where you can, do a complete cerclage. So the moment you start pulling the iris from the periphery, the angle also starts opening up and the pressures also start going lower down. So I think you marry both the worlds. You control the pressures and you control the glare problem as well.
Wonderful, wonderful. I think that seals the area. The contraindications for phakic oil, I think I had covered. Dr. Sonu, we have all covered. Most common complication in my textbook would be probably over, undercorrection, or rotation. Cataract is not the most common complication. I think let us put it out once for clear, because I get a lot of questions even on my YouTube channel about cataract. So in my personal opinion, I don't see cataract so often. I would less than 1% of the cases probably. Your take, Dr. Sonu?
As you start refining your surgery, you hardly see any complications. There would be patients with either a low volt or a high volt.
Absolutely.
And except for these conditions, now cataract is very, very uncommon, because with the V4C model or the central hole, you are just replacing the normal physiology. So I don't think on a longer run, we are going to see cataract very frequently. So raised intraocular pressure could be one of the reasons, one of the commonest findings in the beginners to practice. Or maybe if you tend to touch the iris, you might encounter chaffing and pigment dispersion.
Yeah.
Wonderful.
So
Dr.
I
Himanshu?
totally agree with Sonu. In fact, the only thing what we now see is the size thing part. Right?
Absolutely.
So whatever we do, remember that we are taking a surrogate measurement of wide to wide and putting the lens in sulcus. So there would be some chance of mismatch, and nobody in the world can solve that issue, though whatever we do. So the commonest thing what we talk about is variation in the walled part. And again, it depends on the individual. If anterior chamber depth is decent enough, most of the time you can get away, like Kamal was showing. I have cases where more than 1,000 watt, they are fine deep anterior chamber. But if you have a problem in such kind of thing where the AC depth is low, you might have to change. So size mismatch is the only thing which is possibly the first number of the complication. The rest, everything would be, that itself is also not very common. Overall, very safe surgery, very safe surgery.
Okay, just a caveat here. All the patients where I have a vault, very high vault, 1,000 or 1,100, are ones who are AC depth was more than 3.2. Now that's a caveat for the audience.
Sure.
You cannot get away with a 1,000 micron vault in a 2.8 millimeter AC depth. This is probably sooner or later going to cause a problem. But in case you have a large AC depth, these cases are very, very forgiving.
Sure.
Now, is cataract a common complication? I guess we've answered that. Difference between IPCL and ICL, I think Dr. Himanshu did a thesis on that today. People, audiences, and our fellow friends who have heard him talk can probably go and teach other people. Bilateral or unilateral, and how many days gap? Again, I think I touched that point. Your take on it, Dr. Sonu? How do you operate?
So Sonu, bilateral, okay.
You can answer, Dr. Himanshu, you answer that.
Yeah. So bilateral case, again, it depends on the individual scenario. In our country, in India, bilateral simultaneous cataract surgery is also not an ethical thing. In certain countries, Scandinavian countries, where bilateral simultaneous surgeries are done, well, they can take a call. But in our country, we do not feel very comfortable bilateral simultaneous surgery. And so would be true for the ICL and IPCL also. Also, Kamal rightly said that I want to see how my vault is. Then once the dilatation comes down, though I do hydroimplantation, so my vault, I get to know immediately or at least next day. But still, for the safety purpose, I keep a gap of at least 3 days between two eyes. So typically my 2 ORs, whenever I do, a gap of 3-4 days is there, and I go ahead with the next time. There's nothing right and wrong here. Everybody,
yes, absolutely. I think this is our experience. This is our experience. Dr. Sonu?
My practice is to keep a day's gap between the two surgeries. And on the day one, I would check the vault. And if I'm satisfied with everything, I'll proceed the same day with the other eye.
Sure.
And maybe with the intra-op OCT, we might be a little better off now, where we would in future be able to assess our vault on the table. And then in those circumstances, maybe considering bilateral surgery would be, standard of care. But yes,
wonderful.
The cost factor and everything might be too much to say as of now.
Okay, so we have another question on endothelial counts and its effects by Dr. Devendra and Dr. Jacobson. I think both have discussed that, their studies have proved that if there's no trauma, if you've done a surgery perfectly, it does not matter with the endothelial count. Anyone has anything else to add here?
I think it's fine. It's fine. We can go
Okay,
ahead.
wonderful. Now, how to fill the calculation form for the phakic IOL? I think Dr. Himanshu and Dr. Sonu have both shared about the ICL, and I have shared my form where we use one measure of caliper, one measure of IOL master, one measure of cirrus oblique pentacam, and we also do a biometry. We also mention the mesopic and scotopic pupil size mandatory for all patients. So this is our form, which helps us plan the size and also helps us to plan an IOL at a later date if required. So this is how it would be a good idea to start doing that.
So one clue I would like to give everybody, so most of the companies do not recommend UVM STS measurement. They cannot give you lens based on that. But when you mention that, they actually might have to consider that for their sizing, and they may give you. So if you want a particular size lens, then make sure you only mention what you want.
Absolutely.
So you do not need to write everything, because they get
no.
There is a
I'll tell you again. I will clarify. This form comes to me, and I finalize the size on the side. There's one column which has been finalizing size.
I think that's
so these three readings come to me, and I finalize.
Yeah. So as a surgeon, as Dr. Kamal said, you are the final person who should be saying that, finally I want. Also, what Dr. Kamal said, in fact, we both said same thing, that your caliper is something which I use as my final measurement.
Absolutely.
I take the baseline as my other mearment. Whatever I get on my pentacam, I put it on caliper, and then I go near the eye. And then I can adjust on the caliper. Do I need little more than this? Do I need little less than this thing? That gives me one more measurement. So that's a double confirmation of thing. But final thing, confirmation is by my caliper.
Yeah. Okay, wonderful. Now I think the second last question is how to explant. I think I did. I also have videos of ICL explant, but that would be probably going a bit far away. I've got a lot of videos, interesting videos, but I think there was some problem with the way the presentation was going on. Presbyopic IOL, we've discussed about that. What is the maximum power available? I think it is up to the company to say till what time or what power they can manufacture. I've done 34 with the 2.5 diopter cylinder. I'm not aware of any higher power reported than this, at least to my knowledge. So anybody wants to add something to that?
So just remember that when you go for extreme powers, whether it's hyperopia, myopia or something, possibly Kamal would have done after many years of experience. Right? Whenever you're dealing extreme power, there are a lot of situations which you have to deal with. One, your reliability would be little doubtful because your STS measurement, you might, if you have UVM, surely get an STS done. Also, the lens thickness would be little different. Lens can come really thick and can get stuck in your cartridge. Remember, just this is not the same size as you are injecting.
Your normal lens.
So don't jump over to extreme cases in initial few cases. When you're comfortable with everything, where you can possibly, like Kamal, be confident about every possible thing which you throw at, then go ahead with such cases.
I think that's wonderfully put. And I think a sweet spot for a beginner surgeon would be anything between 7 diopters to 14 diopters is a beautiful play zone, because that's how I let my other people also when I train. Anything between 7 to 14 diopters, a normal pupil size, a normal white to white, a fully dilated pupil. Yes. These are good cases to start with.
Yes.
Now, isolated cases. This is an interesting question by Dr. Sheetal and Dr. Sridevi. Isolated cases of glares and halos, especially if they are sectorial. Now, I think one case which was beautifully discussed by Dr. Himanshu, a patient with microtropia, and another patient, 2 patients I have had is patients were non-orthophoric. They were having fusion problems. And the reason for that was that because once we started doing that aberrometry, and as Dr. Himanshu said, the aberrometry is just measuring the central light in, central light out.
Yeah.
So once we did their orthoptic checkup, we found that the problem was somewhere else. So these cases, especially higher powers, please, I would recommend have your optometrist do an orthoptic test before you take these up patients up for surgery, especially if there's a gross anisometropia. The difference of power between both eyes is different. The eyeball size are different. the angle kappa of these patients would be different. So these patients would have tendency to have glare and halo, especially if they're high powers or high anisometropy.
Now,
anything you want to add, Dr. Himanshu, here?
Yeah, if glare and halos can, so there are cases where theoretically you can have glare and halo from the hole also, and from the material of the lens where it's falling.
Absolutely.
Now, Sonu was mentioning about if somebody has monofocal IOL, I mean a non-toric phakic IOL, and has very high volts, you can rotate the lens upwards. Similar thing you can try before you decide to explant the lens. You can, if it is a non-toric lens, you can rotate the lens. Most of the time, the position of your central hole is never exactly dot in the center. It's always on one particular side. When you change the orientation of the lens, the position of your central hole again changes. And sometimes that itself can solve the problem. But if it doesn't, and if you have to explant, well, that's a thing which you have to keep in mind.
Yeah, I think one more point is a lot of times a large PI, if it is done anything beyond 1 o'clock and 10 o'clock, can also cause a glare. I have had patients operated long ago when we were just beginners. The PIs were big and they were eccentric. They were not being covered by the lid. So we had to go and do a corneal tattooing in 2 of my patients and a Seepser's knot in 3 of my patients, till I learned the lesson to have a smaller PI and always superiorly at 12 o'clock.
Yeah, I prefer laser PI. So that's a very good point, I think, what you have.
Yeah. So I guess that settles most of the questions. And I guess it was a very, very rewarding experience for all of us.
Thank you. So nice. Very nice.
Very nice.
And I must say that thank you to Himanshu and Sonu and Care Group that we were able to have this wonderful interaction. And one thing I would appreciate is all the audience, if their questions were taken care of, how about just throwing me an email or sending a message across on the email which you have and letting us know what do you want to know more in future. And we can promise this Eye to Eye series is going to be here with you. We will touch different topics, have different experts, take your questions the way it was. And I think it was a learning experience for all of us. Thank you, Himanshu. Thank you, Dr. Sonu.
Thank you. Thank you.
Okay.
Thank you. So nice of you. Thank you. Thank you.